This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 651, recorded on August 7th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York State, Daniel Griffin.
Hello, everybody.
How's it going, Daniel?
It's going well. It's going well. We were just starting to talk a little before this started about our big storm that we just had here.
Yeah, a couple of days. Was it Tuesday, right?
Yep.
We had a lot of high winds and rain. You probably had the same thing since you're not far away from us.
Yeah, it was, the biggest thing was you should have seen the sailboats, right? Because the sailboats are all out there and the wind was blowing. So even sailboats that are attached to these huge moorings were actually being dragged around the bay.
Wow.
So it's actually quite something.
Wow. Well, we have no power. We lost it yesterday and it will be out till Sunday, Monday. So I came in to do my my pods today. Got a TWiV in yesterday, and then the power went out, so I had to reschedule TWiVo, and so I came in today for TWiM, and now the Daniel part of TWiV. So,
yeah, all right. So let me start with my traditional quotations. These are both by Winston Churchill. It's a fine thing to be honest, but it is also very important to be right. The second, I modified this a little to make it Winston Churchill says men, but I changed this to people. People occasionally stumble over the truth, but most of them pick themselves up and hurry off as if nothing ever happened.
He was great. Churchill was great.
You really had some great one-liners. I should make sure before I get into the thick of it that I let everyone have an update on our current fundraiser. So I wanted to thank everyone for helping us support FIMRIC. But we've now started August and September. We're going to be helping to support Floating Doctors, which is an organization that I've actually worked with down in Panama. Mm-hmm. It's an organization that I'll say a friend of mine, Ben Lebrat, started many years back. And they're in the northeastern part of Panama. And what they do is they go out by boat. Sometimes it's like 4 or 5 hours across the open water to these tiny little isolated villages where the indigenous people live. And they set up mobile clinics and provide medical care. And unfortunately, because of COVID and everything else, not only are they having more issues with medical problems, the people are actually starving. So it's a tragic situation. I think of all these people that I got to know personally in these small villages, moms, dads, children, and now they're suffering. And so Floating Doctors really needs our help. So if people can go to parasiteswithoutborders.com and donate, we're going to do the same thing. For every $1 donated, we're going to double it to help Floating Doctors. So help us with that.
Before you go on, Daniel, I want to show you a couple of weeks ago, you made a comment. You wanted a Masks Are Cool t-shirt, and there you go.
That's awesome.
Masks Are Cool, This Week in Virology. You can find it at cafepress.com/TWiV. It's kind of a nice design, isn't it?
I want a whole bunch.
Sharon Isern, a virologist down in Florida, designed it. So
this is great. I love this. All right, so we'll start off with where do things stand, and not great. I think as I said last time, my wife keeps saying, "Dan, you're just a bucket of sunshine." We're now past 700,000 people that have died from COVID-19, and many think this is actually an underestimation. And in the U.S., we're still averaging over 1,000 deaths a day. And we're actually seeing a bunch of cases in kids in the immediate area here. And part of it is they're having these delayed like prom parties, graduation parties. And sometimes it's more than 50 kids at these gatherings, and they're all piled together. You see these group photos with people without masks. And so this is this is a bit of a disaster. I'm going to just say that, outright. It's a bit of a disaster what we're seeing. Actually, the last 24 hours for me has been a little bit of a challenge because I think I mentioned I've tried to help with a couple of the programs in the area. And we had some positive tests. But actually, when we retested, it actually turned out these were false positives. So I was going to start to talk a little bit about testing and quarantine and what exactly happens here. Because this is very, as my wife says, it's excessive, but it's excessive because of the biology, not because of anything beyond that. So when initially we were concerned that these were true positives in these different programs, the kids were in cohorts. So you actually think of this almost as a classroom. There's a limited number of kids, 8 kids in a situation where if one of them is positive and shedding virus, you potentially have 8 kids who could have gotten infected. So we'd consider that a high risk exposure. And we have a lot of guidance on this from the CDC, and they actually have a great web page. If you just Google CDC and quarantine, they actually have calendars that run you through this. But let's talk a little bit about, because I think when the ideas of quarantine were put in place, people were thinking about adults. So if you've had an exposure, the recommendation is for 14 days after the exposure, you need to quarantine because you're in the incubation period. If you get a test right away, that's great. You're negative right away. You get a test 2 days later, you're still negative. But anytime during those 14 days, you can become sick or start shedding virus. And here's the challenge, because with kids in particular, they don't necessarily get sick. They don't necessarily show symptoms. So at any point during those 14 days, they can start shedding virus with or without symptoms. Now, that's one thing if you say, oh, this is a 42-year-old man, he's going to be in a separate room, he's going to wear a mask, he's going to use a separate bathroom. But now imagine what we started talking about. You have your 9-year-old daughter, and your 9-year-old daughter just had an exposure. Now, how does quarantine work in a situation like that? It's quite difficult. And I was on the phone last night with several people, and in one situation, it was the mom and the dad. And the mom was like, I am spending the next 14 days with my daughter, period. I'm the mom and that's just what I signed up for. And the dad is meanwhile saying, I'm older. I have some health issues. If our child starts shedding virus and I get the virus, I might actually die. Right. So this is all pretty tricky when you start thinking about now you open a school, right? Which is this is sort of in a microcosm what we're seeing now. And your child is off at school and they come home and say, you know what, just found out one of the kids in my class is positive. So theoretically, they're quarantining for 14 days. I don't know how realistic that is for a young child. One of the parents is going to be taking care of them. Anytime during those 14 days, they might become sick. They might start shedding virus. Now the quarantine restarts for everyone in the household at that day for another 14 days. And it starts adding and adding and adding. And it becomes this sort of never ending quarantine. And we talk about the costs involved in testing people to prevent stuff like this happening. You can already think about the costs of our current model where people are out of work for weeks and weeks. People are getting sick and ending up in the hospital. That's not cheap. We're seeing a long tail and disability with COVID. That's not cheap. Not only is it not cheap in financial terms, it's not cheap in the human impact. So challenges ahead of us. I know with testing, there's actually been a little bit of a push to limit testing, which is actually interesting. I try not to be political, but I was reading an article where they said there was a lack of a national testing program. And my response was, well, we do have a national anti-testing program. So I apologize if that is taken as political. But the way we responded in our local area is we were having the problem that I actually just heard Anthony Fauci talking about, where in certain areas you might perform a test, it might be a week or longer before you get that result. And as he mentions, and that's basically the equivalent of not having a useful test.
And you had such an experience yourself right here, right?
Yeah, we were out to 8 days. And actually that was an issue for us because when we were setting up this, we had the idea that there'd be periodic testing going on. And when we had an 8-day turnaround and we realized that adding more tests to the system was not going to be helpful, we delayed. And then we've actually now gotten our resulting back to somewhere between 12 to 36 hours, depending upon the priority level. So instead of us doing less testing, we said, okay, the problem is that we're having resulting delays. And we've, I'm going to say, at least at ProHealth New York, so all the ProHealth doctors that are listening, congratulations for helping us make this happen. But yeah, we're rolling out we're working with BD to get access to those strips. We're doing Abbott ID now. We invested in a Hologic machine. We're coordinating better, I think, with Quest and LabCorp. And we've got our turnaround time down to about 36 hours or less. So that's huge, but that's a big issue. You do a test and you find out 8 days later, oh, by the way, that child was sick. Just think of the challenges for contact tracing. It's a disaster.
This is where a daily cheap test would be useful, especially in a quarantine situation where you have a kid have an exposure. You could test daily and see when they first start shedding, right?
Yeah. I mean, the nice thing, at least for us with this experience, was we tested, fortunately, they turned out to be negative when we retested them. So they were false positives, but at least the children tested positive. Thank you to the hurricane, we had no sessions. And so the children did not return to camp with, could have been that they were infectious. So fortunately not, but that's what you need. I think the impacts of not doing this, I know our schools in the area, we're waiting to hear what their result is. But I think we talked about last time, there was the comment that, well, if we open the schools, there will be a certain percent of children that will get sick and die. It's only going to be 14,000. Putting only in front of 14,000 children dying is just, not acceptable. We need to do better here. Again, this is not a partisan issue. We all want to, and we all need to do better. So let's work together and make that happen. Masks and eye protection. And I put here, why are we only hearing about this now? So, we've heard about physical distancing. Some people call it social. We've heard about face masks, right? And we've heard about not only do face masks protect others, but maybe they protect you. But there was an article that came out, physical distancing, face masks, and eye protection to prevent person-to-person transmission of SARS-CoV-2 and COVID-19, a systematic review and meta-analysis. And I think now we're getting a little more data on this. I've always, as I think has come up on TWiV several times, I've always included eye protection as part of droplet protection because eyes are mucous membranes. You really don't want people coughing, sneezing, spraying into your eyeballs. And I think now this was a good systematic review and meta-analysis that there is data actually that protecting your eyes is a useful thing. We say not glasses, we say goggles because that's what's studied, but glasses are better than nothing. Contacts, not so great. But I know, I think I talked on one of the last TWiV when my dad is hoping at one point the gyms will open, and I told him only with goggles, a face mask, the full repertoire of protection. But yeah, I think we're hearing about it now because we're starting to get a little bit of data on this. But I'll say actually, as people who work with me out at Plainview Hospital, in February was when I was walking around making sure there was eye protection on all the floors for all the staff, so that this was part of the personal protection that we use there. 4, this is understanding the full impact of COVID-19 by systems. And I like to point out to people, a lot of people get on their computer and they look at the different websites. And I want to say COVID is more than just counting cases and deaths. There was a quotation at one point where someone said, well, with COVID-19, you either live or you die. But it's actually much more than that. A lot of people live, and there's significant, what we term in the medical profession as morbidity. So I wanted to go through each system talking a little bit about what's the impact. Because unfortunately, a lot of people do a little more than just get over COVID-19. And so sort of starting from the top to the bottom, outside to inside, the dermatological impact. So we've talked a little bit about hair loss. So my list of you heard it first on TWiV. And there's actually a little bit of a controversy about what exactly is going on with the hair loss. The initial idea was that it was these are some big words, but we're going to learn telogen effluvium. And this was the idea that after a significant amount of stress, your hair follicles go into a state of really just sort of arrest. They just stop. They're not going to grow. A period of time will go by, and then the hair will start growing back. But one of the things we've actually seen is a significant amount of hair falling out. So some people are suggesting there's a component of anagen effluvium. And this is where you're losing hair during the growing phase. So instead of the hair growing in, it's falling out. And a lot of dermatologists are actually describing that the individuals are coming in with a gallon-sized Ziploc full of hair. It's almost like they brought in a wig. And this is something I think I described in our patient in the ICU recently, where the nurse was saying, hey, look, her hair's all, and the pillow is covered with hair. So it's not that you're losing the normal amount of hair and it fails to grow in. You're actually losing hair at a significantly faster rate, and it's not growing back in. So, this virus just loves to give us everything.
So late sequelae, the hair loss?
The timing is interesting. We start to see this about a month after symptom onset. So, sort of interesting. I think I was waiting for our immunologist to sort out what is going on. The virus is long gone. What exactly is triggering this? And it tends to be, women more than men. So this is my wear your mask now or your wig later. We see multiple rashes, right? Everyone knows about the COVID toes. I have special COVID socks with red toes, my COVID toe socks. But we see all kinds of different rashes. And the COVID toes is something we refer to as pseudo chilblains. And this is as if you were to put your toes into hot water and pull them out and the toes are red. So it looks sort of like that. And this was, I think, people started noticing early on. Nurses noticed it more than doctors. Interesting enough. When I asked doctors, like, oh, I don't know about that. I asked nurses, like, oh, yes. And then they would show me all the different patients. So neurological impacts. So we've discussed many of the acute impacts of COVID-19, which included the confusion, the delusions, the paralysis, the Guillain-Barré we're seeing about 4 weeks in. But now we're actually getting more data on the longer term post-acute COVID impacts on cognitive function. And many of the patients of mine have been reporting issues with concentrating and their attention span. And I actually mentioned one of the local neurologists who's seeing if maybe ADHD medications can be of help because of the shared characteristics. But there was a paper that came out, The Landscape of Cognitive Function in Recovered COVID-19 Patients by ZHOU, Joe et al. In the Journal of Psychiatric Research. And he actually was reporting that, much like was seen in SARS-1 COVID-19 patients who had residual issues with poor concentration, memory, and other neurological issues, this seems to be the case in many of these patients and actually corresponds with the CRP, the C-reactive protein. So the amount of inflammation that we're seeing early on may have some sort of predictive value with regard to the cognitive function that we see later on. So sort of interesting. Couple of reports on a possible link between drop foot and COVID-19, so peroneal nerve palsies. So plenty there in the neurological department. Cardiovascular impacts. So there appears to be a risk of acute myocardial issues, so acute issue with heart failure, arrhythmias. But we're also seeing evidence of longer-term myocardial inflammation. So there was the paper, Outcomes of Cardiovascular Magnetic Resonance Imaging in Patients Recently Recovered from Coronavirus Disease 2019. And that was in JAMA Cardiology. I didn't know there was a JAMA Cardiology. But they actually were showing that the majority, about 78% of the patients, month or two out, we're actually showing inflammation of the heart. So pretty high, significant amount there. And I don't know if people in the news, if we have some baseball fans, the Boston Red Sox pitcher is out for the season due to COVID-19 related heart issues. So this is the Red Sox lefty, southpaw, Eduardo Rodriguez.
Yeah, we've talked about athletes and how you really need to be careful, because a little impact on your performance can make all the difference. And this is much more than a little impact, right?
Yeah. Yeah, this is, and it's really difficult. I think, if you think about an athlete, the risk for an athlete, I think a lot of the leagues think about, all these cases, we may have to shut down. But I think about it on the human level. Think about an individual who has worked this hard to get to that point, and their dream's gone, right? I mean, they have, we don't know. I want to be honest here. We don't know what the long term and whether these people will be able to make full recoveries. But it's a frightening prospect as a professional athlete.
That's why it's probably not a good idea to have Olympics next summer.
Yeah, that seems like a bad idea. Pulmonary impacts. There was another, and this was about 2 months post-recovery from COVID-19. And this was persistent symptoms in patients after acute COVID-19. And, the majority of previously hospitalized patients are still reporting trouble breathing and fatigue. And there was another article, Impact of Coronavirus Disease 2019 on pulmonary function in the early convalescent phase. And they're detecting imaging pulmonary functioning abnormalities in the majority of patients a month after discharge. And, we're seeing cases, publications, severe fibrosis. I see these individuals, they survive, but you do a CAT scan of their lungs and the trachea is pulled over to the side with this traction from the fibrosis. And I think people probably remember, or you had an individual undergo double lung transplant. So the pulmonary impacts, I think, are pretty significant. Vascular thromboembolic impacts, well established, increased risk of pulmonary embolism and thromboembolic complications. So, these can lead to death, stroke, loss of limbs and digits. So pretty significant issues. The gastrointestinal manifestations, probably some of our providers, and I know if you sort of look at some of the COVID support groups, not only can there be nausea, diarrhea, intestinal and organ ischemia as some of like the acute issues, but there can be a chronic diarrhea that lasts for weeks to months. So that's actually sort of plaguing a lot of individuals. Kidney damage. I think we talked early on about this sort of progression from pulmonary to cardiac to kidney damage. But sometimes this is severe enough that the patients are actually requiring dialysis. And actually, if you look at different series, in some series, this may be up to a quarter of hospitalized patients are having some kidney impact. And it's even higher when you look at the high-risk patients that end up requiring intensive care support. And some of the autopsies on the individuals who did not survive are actually showing acute kidney injury with acute tubular necrosis, with lymphocyte and macrophage infiltration. So we're actually seeing the immune cells invading in, and once you see renal failure, actually your risk of mortality goes up significantly. The other thing, I'll just sort of, you'll if you take care of enough of these patients, you'll start seeing conjunctivitis in a few percent. We say it's about 1 to 3%. It's not common, but it's actually, I often see these patients and they've been, given different eye drops. They've been treated for their bacterial conjunctivitis, but this is often just part of this. And if you look at the eyes, there isn't a sort of a thick purulent, it's a watery discharge that we're seeing in these individuals.
And this is early on or later on?
No, we see it at all points, but it's usually later on.
Okay.
Yeah, this is usually part of we'll say the long hauling part of the COVID-19.
Okay, so I want to make it clear, it's not reflecting entry of virus in the eye and conjunctivitis there, right? It's later.
Yeah, I don't think so. Okay. I really don't. It'd be interesting, right, if there was the idea, I'm going to say this is not the case, that you failed to wear your goggles and the COVID got in your eye. Now, that's not what we're seeing. This tends to be a later inflammatory post-viral part of the COVID-19. So, all right. So those are my updates.
All right, we have some questions for you as usual. I always have to pick some because there are a lot, Daniel.
Okay.
First one from Charles, who wants to go back to TWiV 648, where you talked about tocilizumab, and you said, we give tocilizumab without steroids. IL-6 shoots up from the 40s to over 3,000, right? So you're blocking the receptor, which creates this problem. And so Charles asks, okay, tocilizumab binds the receptor. What about siltuximab, which binds IL-6. Could that make a difference together with tocilizumab?
Yeah, I don't know. I mean, these are the great sort of questions that people come up with that sort of help guide us forward. Yeah, I mean that's an interesting. So I don't know if people. So the MAB means these are monoclonal antibodies that we're talking about here,
Mm-hmm.
and siltuximab is an anti-IL-6 monoclonal antibody. It's actually a chimeric mouse one. And so it's made from human mouse. But yeah, it binds directly to IL-6. So it's interesting. Yeah, that's an interesting thought. And in many ways, it makes more sense, right? And I think the reason that we started using tocilizumab was the experience in the cytokine storm of some of our CART therapy and situations like that. So this is probably not being tried by us just because of a lack of experience, availability, the rest. But it makes so much more sense when you think about it. Why don't we just directly target IL-6, neutralize it, rather than blocking receptor with the potential result?
Okay. Then we have one from John in Cambridge, England, who presents some numbers about the weather, which are quite puzzling to me. So you could read them in the show notes.
Yes.
All right. So the first is about sensitivity. And remember, Daniel, last time you said, I'm going to challenge the concept of sensitivity. And he says he agrees. He also wants to challenge the concept of specificity. If specificity means not indicating the presence of viable virus when no virus is present, the clinical PCR tests have poor specificity if their results are interpreted as indicating the presence of viable particles, or in other words, infectious. In the same way that we would give arbitrary sensitivity to Abbott ID Now by our choice of reference test samples, we could give a clinical RT-PCR test an arbitrary specificity when tested against a cell culture reference. So again, specificity in the established sense makes little sense. What does specificity mean, Daniel, when we're talking about these tests?
Yeah, these are great. So we've spent a lot of time, and I think this is good, that people understand. So sensitive would be the question of, if it's there, can you pick it up? And specific is the relationship of, when something is positive, can you trust that? Is that, is it only going to turn positive if the person has COVID-19, if the person only has SARS-CoV-2 RNA in their system? So if something was not specific, it would, any of the coronaviruses will turn it positive. That would lack specificity. So the things you bring up here is, I think, great. So as far as sensitivity, right, there is an issue. It's the PCRs are, as we've mentioned, too sensitive. People who are not infectious are being told they're positive. They're positive in that they have, maybe 80, 90 little bits of RNA, but what we really want to know is, are they infectious? So they're overly sensitive, but yeah, they actually have an issue with specificity. So I like this argument here, because when someone goes and get tested, you can't come into the workplace because you're a PCR positive. The assumption is, you must be infectious. The test has horrible specificity for determining who's infectious. The antigen tests are better in terms of specificity. They're going to pick up a lot less non-infectious people.
Okay, so then he has two comments on units. He said, when you talk about IL-6 goes from 40 to over 3,000, he wants you to say what units.
Yeah, so these are picograms per, I think it's picograms per deciliter, I think, is the units that we're using. Yeah, remember Michael, who was working on the bivalves in the lab?
Yeah.
He used to always, you clinicians, you just use numbers and you never give us the units. I think it's because there's so many darn units that we're just like, after a while, we're just referring to like this. But then it's an issue if like we've got someone listening in the UK versus someone down there in Houston. The units are different.
Yeah.
So yeah. And actually, I should say, like, there was a little comment about like creating a table, and actually we put that table out in the last. So Parasites Without Borders puts out a monthly newsletter. So if people go to Parasites Without Borders and sign up for that, they can actually get that monthly newsletter. And there's one specifically for clinicians, which a little bit more than goes out in our normal newsletters.
So you Can you get the back ones with the table in it, or is it table in every one?
You know what I'll do? I'll make sure that the next big one that goes out, we put that in there. Because a lot of people have asked me about that, and I've started including that in some of my talks. Is it, sort of as you saw that? I wonder if he did this on purpose, that EGSC, and then had a whole thing there, and you're like, what is he talking about? Can he translate that into something intelligible? Maybe that was a clever way of saying, let's, we say picking up 90 bits of RNA, CT39. We're trying, I think, here to translate our testing into something that makes sense to people. You're infectious when you're in the millions. Our rapid, cheap tests are picking up 50,000 or more. They're plenty sensitive. They're more specific for picking up people who are infectious. So this is it's excellent points.
Yeah. So both John and several other listeners had written in asking for that table. So parasiteswithoutborders.com, sign up for the newsletter and you'll get it. It's in there. Is that correct, Daniel?
Yes. Yeah.
And John also wanted to know, when you talk about pieces of RNA, what you meant. Pieces.
Yeah. That's really what we mean. So we mean, so the A's, the C's, the G's, the U's, basically these little sequences, and they're fragments where, when we do these amplification methods, these nucleic acid amplification tests, we're actually looking for small, 120 or less base pairs. And the reason we do short ones is because anyone who's done PCR in the lab is familiar with the 3 cycles. So you heat it up, and that's when you separate. Then you drop it down to an annealing phase, and then you have a midpoint, which is your extending. But with a lot of these PCRs, a lot of these amplification approaches, if you use a short enough sequence, while it's annealing, it'll actually quickly finish off the elongation. You can remove the 3rd phase, so you can have a 2 cycle, and you can speed up your amplification process. So these are little short pieces, and they're not testing for a viable virus in any way. All they're testing is for little bits of RNA, 120 or shorter in length.
All right, one more from Dr. Mary Bowden, who is at Breathe MD in Houston, Texas. I'm a solo practitioner in Houston, have tested almost 6,000 people for COVID since March, primarily saliva through Microgen DX. Are you familiar with that one, Daniel?
No, not really. No.
My facility is unique in that I've been testing asymptomatic people from the start. I've never restricted who gets tested, and since the lockdown ended, I have tested many people who need clearance to travel or go to camp. I'm in the process of analyzing my data, but one trend that really stands out is intrafamilial transmission or lack of it. When I look at many families I've tested, I cannot find a single family where every member tests positive. I have faith in the sensitivity of my test. I just finished a 6-week run of 13% positive test rate, and I understand that T-cell immunity is part of this. Wondering if you could direct me to research in this area, as I'd like to explore it further.
Yeah, no, this is interesting. Dr. Bowden brings up a few interesting things here. So I quickly Googled the Microgen DX, and they're actually using a DNA sequencing to accurately identify COVID-19.
Wow.
And so that's another. We got so many bits of technology out here.
That would be PCR followed by sequencing, probably, right?
Yeah. The couple things that, and we see this too, where we'll test a whole family, whether it's our nucleic acid amplification testing approach or whether it's serology, and you'll see 80%. That tends to be a number we see quite a bit, where 80% are positive, but you've got someone who's negative, and you wonder, did this person just not get infected, or did this person get infected, but they're one of these people without a serological response, for instance, if you're looking at serology testing. And so I know this was actually, I think, a conversation I mentioned I had with Jay Berger, the head of our pediatrics. And he was saying we would love to start looking at T-cell immunity, doing some of these T-cell assays to get a sense, is maybe there are just people who don't respond as far as the antibodies go, but maybe they'll have a T-cell response. And as I think was discussed on immune, there may be people who really just do neither. There's tends to be a type 1 with well, I don't know if I'll get the numbers right, but who have a nice T cell response. There's another group that has a nice B cell response. There's a third group that just doesn't seem to care. Get the virus, goes away, not much of an immune response at all. And those people may be the people who do the best.
I mean, I'd be surprised that there isn't a single family where everyone's positive, but to have many where not everyone is not surprising to me at all, right? And I think if you look back at some of the early clinical reports out of China, there were examples of very common intrafamilial transmission, but not always everyone. So, Mary, you can find those. I can put some links in if you'd like to see those, because we did talk about those. All right, Daniel, that'll do it for another clinical update, COVID-19 with Dr. Daniel Griffin. Thanks a lot, Daniel.
All right. Thank you so much. You take care.
Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello there, Vincent, and everybody else.
Welcome back. You survived Isaiah, or however you say it.
Yes, we did. Although we had our power out for 2 days, that's fine.
All right. A
little stress in your life in addition to all the other stuff. Yeah. Who cares?
You're retired. You don't have any stress.
Come on. I still have to breathe in and out, don't I?
Also joining us today from Madison, New Jersey, Brianne Barker.
Hey, it's great to be here. Fortunately, all of the power stayed on here.
Nice.
So that was nice, and it is 79 and a little overcast here.
Oh.
From Western Massachusetts, Alan Dove.
Good to be here. And in a strange twist, most of our town lost power, but we kept it.
Aha!
Yeah, that's what happened.
Now you know where it went.
Really strange.
Our whole section of like our street and everything is just covered in trees, and I thought, oh gosh, we're going to lose power for a week.
Sure.
Our power is still on, and people who have a lot fewer trees, they've got, the few trees they have are down over the wires, and they're finally getting them reconnected now.
So the storm obviously came over you. It went inland and
yes.
went over.
It went. Yeah. It clobbered us. It was really high winds here. It was beautiful today. Clear sky. Well, kind of. It was storming a little today, and then now we've got clear skies in 81.
You know what was weird as that storm swept through? When you could see across the street, pigeons were out flying. What the hell were they thinking? I mean,
they weren't, but were they flying on purpose?
One never knows with a pigeon.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. Hey, 98 degrees. I'm sorry. That was wrong. 94 degrees. Headed for 98. Sunny skies. COVID still leveled off at a modestly high level. And I did 26 years in Florida, so I have no sympathy whatsoever for you storm wimps.
Right. Rich.
Just suck it up, okay? Deal with it.
The death rate, though, is fairly low, both in California, Texas, Arizona, and Florida. Any explanations, or is that going to happen later?
Don't know. I don't know. I don't imagine any. I don't see anything on the horizon that would portend some change in the death rate necessarily. I mean,
here's all the young people catching it, giving it to older people, and then they die.
I think we're at a steady state relative to that, as far as I can tell.
Okay.
And so we'll see what I think. What actually. The big question is what's going to happen when schools open, if they open. Actually,
some have in Georgia.
The Austin Independent School District just made the decision last night to put off school opening for 3 weeks and make it virtual for the first. I don't know how long, but it's going to take them. I wouldn't be at all surprised if they were virtual for the first semester.
About 15 minutes ago, I got an email from the school superintendent. They've been going around with this. I think I mentioned last time they're talking about doing the hybrid model. And now after the school board weighed in on it and a whole bunch of pretty much everybody in town weighed in on it, because this is New England and that's how we do things. They've decided that they are going to initially start out all online, and then they're going to phase in a start of this hybrid thing, first in the elementary school, then in the middle school, and then finally in the high school. And I guess we'll see if that actually happens. I very much hope that it stays online.
I agree with you.
All right, we have a guest today. She is a partner at Covington and Burling Denise Esposito. Welcome to TWiV.
Hello,
Thank
Denise.
you, Vincent. Hi, everybody. It's great to be with you all today.
Denise told us she had, in preparation, watched the Tony Fauci interview, but we didn't have 10 minutes of banter before that one.
No,
We didn't.
We only had 30 minutes with him, and so we thought we should get right down to business. So that's what that's about, Denise. We want to talk with you about getting drugs and diagnostics approved today. But first, I wanted to find out a little bit about your history and how you got to where you are today. Where are you from originally?
So originally from Long Island, I grew up in New Jersey.
Really?
Yes, I did. I went to Rutgers undergrad, very proud of my alma mater because
All right.
they have a saliva diagnostic test.
Yes, absolutely.
under EUA. Then I went off to the Midwest for law school, and then I landed in 1992 in Washington, D.C., where I've been a pharmaceutical lawyer ever since. And so it's been I've been in Washington for 28 years. And I've been at Covington running the FDA practice for 5 years. And before that, I was at FDA for 5 years. So when I was at FDA, I was on their medical countermeasures team. So I dealt a lot with emergency preparedness. And prior to that, I was actually the general counsel of a biodefense company. So we were in the business of developing vaccines and therapeutics to all kinds of pathogens. They still exist, and they're, of course, now working on COVID. So.
Yeah.
Now, you made the point of telling me that you're not a scientist yet. You're doing sciencey things, right? So how did that come in?
So I was a scientist as a kid, but that stopped at organic chemistry in college. I thought this is a terrible idea.
A lot of people start at that level.
So I was a pharmacy major at Rutgers in pharmacy, and about halfway through my first year, which coincided right around the beginning of organic chemistry, I decided that I liked the business angle of pharmaceuticals, so I didn't necessarily have the scientific brain. And then I went off to law school and became really interested in pharmaceuticals. So how FDA reviews and approves them, how the world envisions them, how people who use them think about the benefit risk, particularly in the context of product liability risks. So I got very interested in all angles of product development. And so while I'm not a scientist, I work with scientists all day. And I'm always saying, I feel like every day I say, all right, pretend I've had 10th grade chemistry and start over.
Well, that's an advanced degree in most people's mind.
So you mentioned that you're currently part of the FDA practice. So does that mean that you work with companies to help get FDA approval, or am I misunderstanding?
No, Brianne, you got it exactly right. So I co-chair our food and drug and device practice at Covington. And so my job is to work with clients who are developing diagnostics, drugs, biologics, including vaccines, and make sure that their development strategy aligns with FDA's authorities. So we have a statute and regs and FDA policies. And then also, if they hit snags along the way, to help them resolve disagreements with FDA, which can be done in a variety of ways. But that's what I'm doing. I'm really working with companies that range from emerging companies that don't have their first product on the market yet to big pharmaceutical companies who are developing something novel where it's kind of a first across the goal line kind of thing. And so there are novel legal and regulatory issues.
Now, so I know you probably can't name specific clients, but are you getting a lot of interesting work from COVID-19 stuff?
We are. So really starting in late January, early February, when things got really bad in China, we had a few clients who worked very early to get drugs over to China for trials. So I've worked with many companies developing antivirals, monoclonal antibodies. I'm working with some companies that are developing the hyperimmune globulin products. So, obviously there's nothing that's gotten across the goal line. Remdesivir is the only one with an emergency use authorization on the drug side. But we have been working around the clock with drugs now, a lot of diagnostics, and now really starting to get into what the regulatory pathway is going to look like for the vaccines.
So has the tsunami wave for hydroxychloroquine died off in your end of things?
Ooh, that's such a sensitive topic. So
remember, she's in Washington.
Have I said something wrong?
Well, I did not work on that one. Although I do have some clients who made it. I think that one came mostly from, in my personal opinion, from the White House press conference. And that, of course, has been withdrawn. And if you look on FDA's website, you can still pull up the paperwork, and it has a big withdrawn sign across it. So,
right.
But, I think one of the interesting things about that is, and you might have seen that FDA Commissioner Hahn wrote an op-ed in the Washington Post a couple of days ago. The FDA is a science-driven agency.
Absolutely.
But the commissioner is a political appointee who reports to the Secretary of Health and Human Services, who's a political appointee, who reports to the president. And so you're always dealing with the issues of FDA fending off political pressure and really letting the science drive the decisions, even when there's enormous public and political pressure. So I think hydroxychloroquine was, from where I sit, a rush out the door, and then FDA looked at the science as it evolved and said, we're not going to support this.
When you were chief of staff, who was the FDA commissioner at that time?
I was the chief of staff to Margaret Hamburg.
So what kind of things did you do in that capacity?
So anything that comes up, I think that the range of, I'll tell you the highs and lows, everything from dealing with the Ebola crisis and trying to get diagnostics under EUA for Ebola. So, really interesting, important things. I got called one day to deal with what was believed to be, but thankfully wasn't, an actor-shooter incident on the FDA campus outside of Washington. Turns out that doesn't really align with my skill sets. I think a lot of
that's reassuring in a way.
Yeah. Ensuring that Commissioner Hamburg's, all of her priority issues were taken care of, communicating with the department and the White House on key issues that are going on of interest, making sure that her her relationships with the members of Congress are maintained, and if there are calls from the oversight committees, we get them on the calendar, we prepare her for them. Working with her to manage what's going on in each of the centers. So FDA is a big organization, but in my world, the three centers are devices, biologics, and drugs, and it's a big job for her to oversee all of that. So ensuring that she's up to speed on the important developments in the center, that she provides whatever support the centers need. So it changed every day. And it was, being chief of staff in any of these government jobs, you come in the morning with a to-do list and you leave at the end of the day and you've not touched anything because it was
it's not a to-done list.
But you now have a new to-do list.
Exactly.
That's right. A to-do list.
The crisis of the day.
If I'm not mistaken, Hamburg was a health commissioner in New York City, right?
New York City, that's right.
He was.
Right.
Yep.
That's right.
So basically, if FDA was a ship, you were the first officer.
Yes. Some days I had less important jobs. Who are the people that take the water out of the bilge? Some days.
That's also pretty typical of a first mate who. Yeah,
that's right.
Yeah. Some days.
Now, you were an employee of the government employee at that time, right? In that position?
I was a government employee. So I was actually not a political appointee. I was there during the 5 years of the Obama administration, but I came in through the Center for Drugs. I worked for Janet Woodcock for a couple of years when I came in, and I was doing regulatory policy work for her. Mm-hmm. And then I went over to the chief counsel's office, and I was the deputy chief counsel for drugs and biologics. So I had the staff of lawyers who supported the Centers for Drugs and the Centers for Biologics. And then I was the chief of staff, and so I saw the agency from the various angles. But yeah, I was an employee for that whole 5-year period.
And then you decided to go back into private practice after that. Why'd you do that?
So Commissioner Hamburg stepped down in April of 2015, and I got an acting commissioner up and running, and we brought in a deputy commissioner, Rob Califf, who ultimately was confirmed as commissioner, and I was pretty much ready to get out before the election, which in hindsight was actually an excellent decision.
Indeed. All right, so let's talk a little bit about approvals of drugs, vaccines, diagnostics, and how it's different in these pandemic times, right? So let's start with a drug, an antiviral drug. What would be generally the normal process?
So the normal process for an antiviral is that you'd have to meet a standard that persuades FDA, and there's a statute that governs this. So the statutory standard is that an antiviral is in a class of drugs that gets approved under the Food, Drug, and Cosmetic Act, and you have to demonstrate substantial evidence of effectiveness. That's kind of a term of art, and then safety. And FDA historically, the position has been that really means 2 adequate and well-controlled clinical trials. So we're talking longer term, randomized against either a placebo or standard of care, probably 3 to 5 years in the clinic, and possibly some post-market work. And you have to that all gets submitted in a very substantial package, like a room full of documents under a new drug application. And then FDA has...
I've heard it described as tractor-trailer loads of documents.
Yeah, absolutely. And now it's whatever the electronic equivalent.
Yes, it's terabytes. Yeah.
It's actually dumpsters full of...
Dumpsters now get uploaded through a gateway.
Yes.
And then FDA, so the company, the sponsor, and NIH and other hospital sites, including yours, I'm sure. So everybody's involved, and you have all this clinical work, and it all gets packaged up, and then the CMC, the chemistry, manufacturing, and controls work gets done, and FDA inspects, and then FDA has, under normal circumstances, about 12 months to review that. So you submit it, and they have 60 days to decide whether to accept it, and about a year. And so we're talking about a good year before anything gets out of the FDA. They can do it faster. There are priority reviews that take 6 months. But obviously in a pandemic, none of that is going to work. So I guess on the usual process, best case scenario, an infectious disease in oncology, we're seeing things get on the market 3, 6, 8 months after the application is submitted. But the norm for most drugs is really a year. So that's what we're looking at in normal course.
It's the same essential process for vaccines?
Yes, it's interesting. So drugs that are regulated as biological products, so anything that meets the definition of a biological product under the Act and FDA's regulations, which again, I'm not a scientist, so I call them small molecules and big molecules. The big molecules, which have a certain length of amino acid chain or some crazy thing like that.
That's very good.
Right? Those get approved under a different law, under the Public Health Service Act. And for biologics, this comes from the old Public Health Service Act when NIH used to approve biologics. The statutory standard is safe, pure, and potent. So potency was really efficacy. So now, however, FDA has kind of merged those standards. And for practical purposes, the same standard and the same timelines apply for vaccines and therapeutics, vaccines being sort of subset of biologics.
But you said that even before FDA gets the material, it is 3 to 5 years of clinical trials, essentially, right?
Yeah, I mean, a long time of development, starting from drug discovery, to the bench, to non-clinical studies, to human clinical studies. And of course, as you know, the human clinical studies are done typically in 3 phases. And those phases often don't overlap. Sometimes in oncology, particularly, they'll overlap, but typically you've got 1, 2, and 3, and those are serial, and those could take many years.
So and to be clear, this is not a process that's designed specifically to be slow. It's a process that's designed to be careful.
Right.
And that the result of that is that all of this stuff takes this much time.
That's right.
Right. And that's exactly right. And, I'll talk a little bit, I'm sure Vincent has a question for me on what happens in a pandemic. There are different authorities to put drugs on the market under temporary authorizations. But even then, it's got to be driven by the science. So you have to have some evidence. Otherwise, you end up doing more harm than good.
So vaccines and antivirals are roughly going to be in the same timeline, right? 3 to 5 years, and then 6 to 12 months of FDA review. And of course, we remember back in the HIV outbreak in the 80s, right. There was fast tracking. That was the first fast tracking I remember right, they said we have to get drugs out. And in fact, it was pressure by the community that did a lot of that, right?
That's true.
How about diagnostic tests? Are we on the same timeline?
Diagnostic tests are a completely different ballgame, much, much more quickly developed and much more quickly reviewed, because the review process for medical devices is under a different statutory provision, and they're not reviewed and approved. They're typically cleared. And a lot of the work some of the work is submitted to the validation studies are submitted to FDA, but a lot of the work goes on behind the scenes. And a lot of these companies, the manufacturers and laboratories who have tests that are already on the market, can very quickly figure out how to convert them with varying levels of effectiveness to a new pathogen. So we see those coming out very quickly. And if you look at Ebola, if you look at SARS-CoV-2, we've got now over 200 diagnostic tests. Most of them are the PCR tests, but over 200 diagnostic tests, and only 1 drug under EUA.
Right. So
I gather some of the reasoning behind that regulatory divide between testing versus other things is that there's less harm that you can do with a test. I mean, a bad test could have secondary effects, but it's not like you're going to get a bad test result and die as a result.
Right. I think that's why FDA in emergencies has gotten EUAs out much more quickly. First of all, the need is urgent, because you've really got to try to start to diagnose people very early before you can even treat them. And then from a public health perspective, you need to know where it's traveling. And yes, Alan, you're not, injecting a novel therapeutic into someone. From a public health perspective, if you have a high false positive or a high false negative, it can be hugely problematic. And obviously for an individual patient misdiagnosed, you can have a problem, but you're unlikely to cause, a severe adverse reaction up front.
What's needed to get a diagnostic approved? So you said 3 to 5 years for an antiviral vaccine. So what do we need? How much time and what results do you need for a diagnostic?
So for a diagnostic product, it varies whether you're doing a molecular test or if you're doing I'll just divide them up. For example, for SARS-CoV-2, you've got the PCR molecular tests. You've got 2 different kinds of serology tests, the antibody test and antigen test. And they all have slightly different demands in terms of what you have to provide to FDA. But if you think about the whole construct, you really have to prove that from sample to readout of the result, every step is validated. And so depending on the different sample types and the different technology on which you're running the test and the different equipment, different sample handling techniques, those are all going to have to be validated from soup to nuts to make sure that you're getting the correct test result at the end at a very high level of success. So somebody, in normal times, FDA wants you to be in the high 90s, but right now we're seeing products out there that are probably around 80% effective. But the key there is validating the scientific methodology.
So if you're the serological test, let's say for epidemiologic surveys, who cares if there's a 2% difference in the positivity and negativity? But when you're trying to tell if someone has actually had this infection or not, they came to our town of Fort Lee, New Jersey, and gave 2 tests for free. They were testing the Rutgers spit test, which was great. We got the results back in a day and a half.
Right.
We got no results back from the serology because it was not an FDA-approved test. And when we asked them if it was, they said, we don't know. So that's crazy, because I think
it's got to make you feel good that they don't know whether the test.
Sure, it made me feel helpless, as a matter of fact, because then I started to have a conversation with them, and they gave me the same excuse that you did, Denise. I'm not a scientist. I'm a phlebotomist. So that's
better than a lawyer, but not by much, right?
Yeah, well, they both demand blood in a certain way.
They bleed 2 different ways.
That's right. Yeah. So this is a bad time for all these jokes, but I was very disappointed not to get back an antibody result. And I think it's because the test was poorly chosen, and yet they were still willing to draw blood, which is a small risk, but nonetheless a risk. And they were willing to send it out for diagnosis. So what role does the FDA play in preventing subpar tests from being used?
Yeah, it's a great question, Dixon. So FDA, their mandate is to protect the public health, right? So they definitely have authority to regulate these products. I think what we saw at the very beginning of the pandemic was that FDA was perceived as holding up these tests, and that, laboratories were developing tests, academic institutions were developing tests,
that's right,
manufacturers were developing tests, and FDA was saying, we haven't looked at these tests, we don't know if they work, and you can't use them, you can't launch them, you can't market them. And so there's a lot of political pressure on FDA. And so FDA put together an enforcement discretion policy, which means even though it's against the law to market and sell a diagnostic test that's not FDA cleared.
If we see any illegal tests, we will enforce the law. Yes.
Enforce the law. You can report that to FDA. But what FDA did was they announced this policy that said, as long as you self-validate these tests, you can put them out on the market. And then after you put them out on the market, you have 15 days to submit your application for an emergency use authorization. And it varies by test type. But in broad strokes, then the concern was they swung the pendulum too far in the other direction. All of these tests were getting on the market that really had no regulatory review. So for really good organizations, like the good hospitals, the good laboratories, you expect to have good science and responsible use of the test.
That's right.
But then there are a lot of opportunistic organizations who also said, well, we can do this, we'll put something out there, and they turned out not to be good. So FDA has now developed a system where they pull things off pretty quickly. They look at the validation studies, and if the science isn't good, they pull them off. And there's a list of products where companies have notified FDA of the launch of a test. And then that list gets smaller because FDA takes them off and puts them on the list of tests that were on the market and now aren't. And FDA also, they just did it. I just saw an announcement last night or this morning. They can actually withdraw EUAs. So if an emergency use authorization has been granted, but once the test is out in the market, the performance isn't what FDA expects, they can pull the EUA.
Which was what happened with hydroxychloroquine, right?
Exactly.
So what does validation consist of?
Oh, Rich, this sort of plumbs the depth of my engineering knowledge. So
it's on page 43. Come on, Rich.
Yeah, there is guidance on this. So this is my layman's description of this. So it's everything from how you collect. So let's take a patient anybody. It doesn't even have to be a patient. It could be someone who shows up for workplace testing, and they're going to give a sample: saliva, anterior nares, deep nasal, whatever it is. And then that sample is going to be put in some transport tube with transport medium. It's going to be shipped. It might be stored, and then it's going to be run on I skipped a step. It will be prepared to be run and then run on an instrument, and then a result will come out. So from a scientific perspective, every step of that needs to be validated and proven in different ways for the different pieces of technology that touch it along the way. And then you have to provide all those results. So some of it is sample handling, showing that a test showing that you preserve the sample. So from the time you took the sample to the time it arrived at its location, it didn't degrade. And so there's a scientific method that I don't understand for that. And then for each step, you have to show that you have an accepted scientific method. I think I'm moving around too much because my camera just got...
Your camera just went blurry.
Come on, refocus.
It's not because our eyes glazed over.
Focus.
Well, yeah, are you glazing over?
You're avoiding the question here.
Right. So each step has a scientific methodology. So the equipment being, sufficiently calibrated and appropriate to take that test. And then if you vary from that, FDA works with you on bridging studies. So for example, if you wanted to go from an anterior nares sample to a saliva sample. And so FDA engineers and chemists speak to the company's engineers and chemists, and they come up with a validation plan. And then that's conducted, and that, if FDA accepts it, then they can issue an emergency use authorization.
Does this have to do at all with comparison to some known gold standard test?
So what they've done for diagnostics in an emergency setting is it gets compared to the first test that they've approved that they have a high level of confidence in. So, and then FDA and CDC and NIH and others have tried to develop these banks of known positive and known negative samples.
Sure.
So if you know that there's a test that has a high level of reliability, those get run. And then, so let's say you have a big, PCR instrument, like a Roche or an Abbott instrument, and they've got a test that works and it works. Then you test your new test, you do, let's say 30 positives, 30 negatives, known samples, and you check it against theirs. And then there's, obviously a statistical method brought to bear to ensure that.
So does that mean that from the point of view of the developer, you actually have samples from this bank sent to you that you then run through your protocol?
And work like that.
So the government, I don't actually know where the samples come from. I know FDA doesn't maintain them. They have their own laboratory with some samples that they test against. But I understand from clients of ours that they've had trouble getting the samples. Unfortunately, it's gotten easier because of the high rate of positivity in the country. But at the very beginning, with everybody doing a lot of research, it was hard to get enough samples.
So, Rich, there's a College of Pathology that's overseeing a lot of the laboratory tests that are done in hospitals. And they have a core of samples that they send out to all the hospitals at a certain time of the year. And they all take the test and they send them back, and the laboratory gets certified or not based on their results. So in a parasitology lab, which was my specialty, that's exactly the way it was done, not through the FDA, but rather through a lower court, so to speak, of, in this case, the College of Pathologists, the FRPPC, or whatever those initials stand for. They're the ones that oversee the quality of that test. It was a pretty simple test. You're talking about very complicated things that can rot on the shelves or not be properly acquired or things of this sort, which didn't really figure too much into the parasitology lab, but I'm sure in the virology lab it certainly did.
So Denise, can you give me some idea of how long the process is to get a diagnostic approved, or is it all over the map?
Well, if you look at from the beginning of the pandemic in the US to now, FDA has approved over 160, probably 165 molecular tests. Wow. The first one was a CDC test, which did not have a great experience, and that came out within a few weeks. And I think most of them started coming online really in earnest in April. So I would say that was really a 2-month period from when the work started. And if you were going to do it in the more traditional fashion, you're probably looking at something more 6 to 12 months. It will vary, and the companies that have a lot of experience can do it faster. But I think FDA has done a great job getting these out there. I think under very difficult conditions, the political and public pressure to get them out quickly balanced against the science of ensuring that they work.
So even CDC has to get tests approved, right?
Yeah, so that's interesting, Vincent, because that's kind of a little known fact. Everyone says, well, you're the government, shouldn't you be able to do it? You've got specialists, et cetera. But so FDA's statute applies to CDC and NIH as well. And so, I should probably step back and talk a little bit about these emergency use authorities that FDA has, because, as I mentioned earlier, by statute, FDA has to hold products to a specific standard. For drugs and biologics, we talked about it's, safe, pure, and potent, or substantial evidence of effectiveness and safety. Diagnostics have to be cleared under a different procedure, and these are in-depth processes. But it's by statute, so FDA doesn't have the authority to say, well, we'll just waive that. And so what Congress has done is they have given the Secretary of Health and Human Services, so in our case now, Secretary Azar, the authority to, once he declares a public health emergency, as he did for COVID-19 in January, he can then authorize FDA to issue emergency use authorizations, or we're hearing a lot about these in the news, EUAs. And the statutory standard for approving a medical device, or including a diagnostic or a drug under that, is much, much lower. It is reasonable to believe that the product may be safe and effective in the context of the public health emergency, that the benefit may outweigh the risk. So it's a very loose standard that puts all of the discretion in FDA to decide whether something has a positive benefit risk ratio. And so once that declaration is issued, which Secretary Azar did separately for different kinds of devices, diagnostics, and then later drugs and biologics, then these companies could come to FDA and say, we think we can design, for example, on the drug side, we can design a very small trial, or a trial that might be for a limited patient population. So if you look at remdesivir, they applied for an EUA for a limited subpopulation of hospitalized patients who have been diagnosed with COVID-19. Other companies are developing products for different patient populations. And the idea there with the EUAs is that you could take data from either an expanded access program, which is more difficult, or a smaller clinical trial, and get enough evidence to FDA that they feel comfortable that the drug is going to do more good than harm, and get it out there for people who are gravely ill. And then continue the development program with longer trials, with randomization, better controls, possibly additional subpopulations, and then get it under a full marketing authorization. So that's the idea. It's sort of a temporary fix to get products on the market when people really need them.
And
so you mentioned if somebody has okay, we're all going to talk at once. What's the mechanism if somebody has a diagnostic or a drug or vaccine that has been approved elsewhere in the EU or in Japan or China or Korea or wherever, and they want to bring it to the US, especially under the EUA type of approach, because obviously in China they've been testing, they were testing for SARS-CoV-2 in January, and right. What would have been the mechanism to import that kind of a test?
Yeah. So there are kind of two different legal hurdles you have to overcome to do that. So the first one is you have to figure out how to get the drug under EUA. And even though it could be under an emergency use authorization in another country, or it could be approved in another country, you still have to present your package of evidence to FDA. So that goes in through the application process that I discussed earlier. And then you also have to get authority to import it.
Right.
Right. So, but once you have an EUA, in the context of a pandemic, it's not difficult to get the appropriate authorization to import it, but they're two different pieces. And I think that what we saw a little bit with diagnostics was that there was some concern that some of the diagnostics that were coming in from China and elsewhere had not been validated to what FDA considers its higher standards, and so they weren't able to get under EUA here.
Brianne, go ahead.
So you mentioned, the EUA, and that, am I correct that that applies to mostly things like drugs and diagnostics, and how does that relate to vaccines?
Same construct. So it's the same statutory provision, Section 564 of the Food, Drug, and Cosmetic Act, if anyone wants to read it.
Which is a real page turner, I'm sure.
It is a page turner. So it's the one place in our statute that all kinds of devices, so ventilators, PPE, diagnostics, those are all under that, as well as all drugs and all biological products, including vaccines. So that works the same way. And as you know, there are multiple vaccines that are starting clinical trials. Some of them sponsored by NIH and some of them sponsored by private companies. And there are a lot of global coalitions focused on this, but we envision that FDA would use a similar process where we would start with an EUA and then work their way to the biologics license application down the road.
So the vaccine and antivirals that are in phases, they're overlapping, so that's kind of unprecedented, and everything is accelerated. So how does that get sorted out? Who decides that, and who makes sure that it's still going to go properly?
Yeah. So there are three important things to think about currently. So normally it's FDA. And then we talked a little bit earlier about BARDA, which is an arm of HHS, which procures vaccines and therapeutics for pandemic response. So FDA is the arbiter of the science, and under normal circumstances, will always decide whether a vaccine is safe, pure, and potent. So is the vaccine effective? Is it safe? What are the populations for which it's been proven to be effective and safe? BARDA has an interest because they are, I think, going to be involved in procuring vaccine for the government. And in this administration, they've developed two working groups that are kind of out of the ordinary for us. One is Operation Warp Speed, which you might have read about, which is kind of an operational group, which I think
that's one way of describing it. Yes.
Yeah, we all have our own opinions of Warp Speed, and I've thought a lot about their kind of role vis-a-vis FDA. And then there's also the working group called Active that is sort of reviewing some of the science. But I think what's going to be interesting to see play out here is that normally you would have the data, it would be developed, it would be submitted to FDA, and FDA would review it for safety and effectiveness. And you really wouldn't have members of the Department of Health and Human Services or of the White House or DOD or the VA opining on the science. Here, because we're in different times.
Or tweeting about it.
In multiple ways. Certainly not tweeting about it. There was this concept when Operation Warp Speed was put together that they would, and this is all public. This is out. They put out some PR pieces very early on where they said, well, their hearts were in the right place, right? We want to make sure all the appropriate resources are going to be devoted to the vaccines and therapeutics that win. And so we're going to pick them really early. And FDA said, well, you can't kind of pick them early. That's not the way drug development works because you have to see the data first. And so I think there's a little bit of push-pull there where there are going to be earlier efforts to try to identify the therapeutics and the vaccines with the best data and ensure that manufacturing capacity is there, raw materials are there, vials and syringes are there, and for those products. And so I think what we're seeing is that Operation Warp Speed is going to try to look at the top 3 to 5 vaccine candidates, the top 3 to 5 therapeutic candidates, and at least try to winnow it down. I think that'll be quite controversial because it's not, and there probably will be legal challenges because we have a statute that governs how FDA operates, and within the statutory framework, they apply the science. And so it will be very interesting to see how that plays out. And then active.
And when a lawyer says interesting, they usually mean it will involve a lot of lawyers.
We'll
contentious.
keep a lot of lawyers busy. I think there'll be. We are seeing an unprecedented ability of major pharmaceutical companies and small biotech companies and suppliers and contract manufacturing organizations to all band together and pull in the same direction. However, if Operation Warp Speed were to pick a winner where the science didn't support it, I think you could see legal challenges around whether the government funding went to the wrong place. And I
hope we would see legal challenges around that. Yes.
And we'll certainly see congressional inquiries around that.
I think maybe the word enterprise has a new meaning here.
There was, well, as you said, some manufacturing has already started, right, on some of these and in huge numbers of doses. And as Tony Fauci said on our interview, what we'll lose is money, hopefully not lives, right?
Right.
So a lot of companies started manufacturing at risk even before the government funding started flowing in. And then once the government funding started flowing in, there's really been a lot of manufacturing done. And the other thing that I guess we should talk about that we really haven't seen play out is going back 20 years, right after 9/11, where I think, we had the anthrax attack scare right after the attack on the World Trade Center and the Pentagon. And I think at that point, the government really started focusing on being more prepared up front rather than constantly being reactive in biodefense situations and in pandemics. And so BARDA was created and a lot of government funding was created to develop these products before we need them. And you may know this, there are 3 locations or 3 organizations in the U.S. that have these contracts. They're called CIADM contracts, and the D and the M are manufacturing. And I could probably, sort of centers of innovation and advanced development and manufacturing or something like that. But 3 locations in the U.S. whose job it is to work in public-private partnerships to identify ways to react to particular pathogens of interest. And so these locations are maintained as warm bases for manufacturing. So they turned the lights on at the beginning of the pandemic, and now all of them are ready to manufacture different kinds of vaccines and therapeutics, whether it's, monoclonal antibodies need a different infrastructure than antivirals, and that needs to be different than vaccines. And even within vaccines, you need different types of manufacturing equipment. So those are up and running. And so there's a lot of work already being done, even before we know which ones are going to work in the clinic.
Did you say those were public-private partnerships?
I did, PPPs.
Okay. So, Denise, a couple of weeks ago, the FDA issued a very long document about vaccine approval procedures that, I guess, in part to assure that everything would be done properly, as you have indicated. And that, they said anything above 50% efficacy, I think that was the number, would be approved. And I just wonder why that number was picked ahead of time as opposed to waiting and seeing.
Yeah, so I can't answer that question. I can't answer it because I don't know the answer, not because I'm being cagey. I don't know why they picked 50. So there's been some speculation that 50 is sort of substantial evidence. I actually think that's probably not the case. I think that, I would assume that they've worked with CDC and the epidemiologists to try to figure out what would be reasonable. Obviously, everyone would love 100% or something nearing 100%, but I think that we still don't know anything about immunity to this pathogen. And so I think they had to pick a number that seemed like you're better off taking it than not taking it, as long as the safety profile is acceptable, particularly for healthcare workers and other first responders.
Yeah. It just seems
I read it kind of as like based on experience with the flu vaccine, which is really, if you look at the numbers from year to year on paper, it's kind of a crummy vaccine, right? I mean, you get efficacy numbers for flu vaccine of 50%. That would actually be a decent number for a flu vaccine. And I think it may be based on that kind of an experience at FDA that okay, it's got some efficacy. 50% is a threshold that you could pick, and that's
and it
something
seems achievable.
It seems achievable and is better than nothing.
Well, it's probably based also on the experience that the health community at large has experienced with regards to malaria vaccines. The most protection you can get from preventing malaria is up to something like 60%, but the death rate is greatly reduced in children. Even if they do catch malaria, they'll have a better chance of fighting it off because they've received the vaccine. So that kind of a scenario could play out here very nicely.
And that's another thing that would also fit with the flu perspective.
Yeah, exactly right.
Because the efficacy, I mean, you do these rigorous efficacy studies and you say half the people who got the vaccine got the virus anyway. But if you look at the mortality figures, you see that the people who got the vaccine were a whole lot less likely to end up in the morgue.
Hopefully.
And so
hopefully they will use that as an endpoint, right? Not just infection.
This morning in a different role, I heard a whole basically seminar from a professional in benefit risk analysis. I didn't realize that this was a discipline. Okay. And so, Denise, you must have some experience in this, and I wonder if some sort of, high-end benefit risk analysis wasn't even applied to this situation to come up with some of these numbers.
Yeah, so that's interesting. I don't know the work that's been done internally at FDA, NIH, and CDC to come up with that number, but there is a cottage industry in benefit risk analysts because everything that FDA does is a risk-based analysis. Every drug has adverse events, and the tolerance for risk is much higher when we're talking about HIV and oncology than when we're talking about the common cold or eyelash growth, for example. So there are these companies, and they're combinations of for something like this where you have a broad public health concern, epidemiologists, immunologists, biostatisticians, and there are these very complex models. And so I would like to think that the government was pulling together to figure out what would be acceptable. And look, if one of these vaccines is showing at 90%, and they can fill the capacity, the supply needs at 90%, I think you'll see 50% go by the wayside, and FDA will up the expectation.
Yeah.
This is true. It's
just guidance. I mean, what they put out is just guidance.
So I want to clarify something. This is going back a little bit, but I want to make sure I understand something. You've talked mostly in terms of therapeutics and vaccines in terms of the FDA approval for ultimately approving or licensing whatever it is. But am I correct in understanding that at every step along the way, the different phase trials, you need some sort of FDA approval to clear you to do a trial? So there's oversight all the way along.
Yes, there are human subject protection statutory requirements and regulations. And so before you start any trial in humans, you have to submit to FDA what's called an investigational new drug application, or IND. And once you submit it, the way that works is a little bit different than drug approval, where you have to wait for an affirmative approval. If the FDA doesn't come back to you within 30 days and put your study on clinical hold, then you get the green light to start. And so if you're starting first in humans, you submit your lab work, you submit there's the scientific hypothesis, there's a lab work, and the non-clinical, and then you start your phase 1, which is small and principally safety focused.
So,
I'll stop there, and then if you have questions, then I'll go to phase 2. Yeah.
No, go ahead.
So as I say, the FDA then wants to see that safety data because that's their number one concern in early days, is making sure that you're not doing harm. And then you go to FDA with that safety data, and they work with you to design a phase 2. And then ultimately, unless you're on an accelerated pathway, which there are some, after phase 2, you go back and you work with FDA on a pivotal study design, which is a phase 3 that would support registration and licensure of the product.
So in the case of diagnostics, is there any sort of equivalent process where there's trials of some sort that you have to pass, or some periodic monitoring to make sure that what it is you're doing is, doing what it's supposed to?
Yeah, so there is. It's less focused on human subject protection because you're not administering the product to humans, but you do have to go, if FDA is going to require a clinical trial, a lot of times in diagnostics, it focuses on the instructions for use, making sure that the instructions work appropriately, and that if it's anything that involves user intervention, so home collection or a healthcare provider collection, and FDA requires an IDE, which is an investigational device exemption, then there will be clinical oversight. But for certain technologies, FDA can clear a device without additional clinical work. If it, and this kind of gets into too much of the device law, but certain devices are cleared based on predicate devices that are already on the market.
So Denise, we're at an hour. I promised to let you go after an hour. So let me ask you one more question. A couple of episodes ago, we had Michael Minna on from Harvard, and his idea is to make $1 daily tests, antigen detections, paper strip, lateral flow tests that everyone could take every day. And he lamented that these weren't going forward. And do you know anything about this, and can you shed any light? Is that sort of thing going to happen?
So I think the answer lies in the science. So I know there are companies out there trying to develop very small machines, point of care, and ultimately they would love strips. I mean, if you get on the internet, you can probably find a strip COVID test. I wouldn't buy it. But they're out there. So I know that companies are working on miniaturizing the technology and rapid tests. I think that from FDA's perspective, they would love to see something that's much more user-friendly than a deep nasal swab followed by 5 to 7 days to get a lab result. But my understanding of the technology is that it gets harder and harder to do when you miniaturize it, when you change from having large samples to small samples, and when you change the technology. So I think everybody would love to see that happen, but I think it's going to take some time because FDA is going to be very reluctant and skeptical about the science.
I think the key legal hurdle that Michael Minna's approach faces is that he's advocating doing this type of test, which is actually, I mean, scientifically, these are fairly easy to make, these antigen tests. The chief problem with them and the chief hurdle at FDA is that they tend to be very low sensitivity. So he's talking about tests that might have 50% sensitivity, but he makes a pretty compelling argument that if you've got a test that costs $1 and takes 15 minutes, it's okay if it only has 50% sensitivity because you just go around testing everybody every day. And by the time the test has enough, by the time they overcome the threshold of that test, you quarantine those people. And by doing that, again, he makes the pretty compelling argument that people really should go back to the episode and watch, that we could actually get the pandemic under control a whole lot faster than waiting on a test that's got 99%, sensitivity. He also
makes a point that this is sensitivity compared to the gold standard RT-PCR experiment. Okay. And one of the things he's saying is they don't have to be that sensitive, that in fact, people are not transmissible unless they have such a load of RNA that it's going to be picked up by these less sensitive tests. And the question is FDA going to be open to that kind of thing?
And that's an important question, Alan and Rich, because what FDA is doing as you watch their policies evolve, they've now created a framework where they're thinking about testing in 3 buckets. Diagnostic testing, where you really have a patient at issue and they're not going to waver on the sensitivity. You have to know. Screening testing, where when you're sending people back to work or school, or a venue, they can get quick tests. And then public health surveillance. And I think on the screening and surveillance, they will tolerate a lower sensitivity rate, but not on the diagnostic front. Right. So that's all to be seen, but I think their thinking is definitely evolving on that.
So just on July 29th, there was a release from FDA, new template for at-home and over-the-counter diagnostics for use in non-lab settings like homes, offices, and schools. So they're basically encouraging people to submit these, right?
They are. They've encouraged them. And what's been interesting on the diagnostic side is that every Wednesday at 12:15 p.m., FDA has a town hall on diagnostics. This is unprecedented transparency. You've got the head of the diagnostics division, and he talks about updates with another colleague who runs a subdivision in his unit, and then they do a Q&A. And he has said repeatedly, we're encouraging the science around asymptomatic testing. We're encouraging more studies of saliva samples. We're encouraging home testing. So they're encouraging it, but what we're hearing is they haven't yet seen science that gives them comfort.
I see. Got it. So would you say, Denise, that in terms of all these things we've talked about, approvals and processes, FDA related, is all of this happening now? Would you say it's unprecedented?
Absolutely unprecedented. No question about it. I mean, I think, FDA obviously reacted and was very busy during Ebola, during Zika, during H1N1, during, all the flu seasons, but we've not seen anything like this since 1918 in the U.S. And
we didn't have an FDA then.
We didn't have this FDA at all, right? So this is unprecedented, and they've really pulled in a lot of resources, and they've reallocated resources, and it'll be interesting to see the effects on drugs and devices that aren't for COVID, because I think we're seeing slowdowns in those areas, but FDA has really stepped it up.
Are there any changes as a result of this pandemic that FDA will make to speed things along and still remain efficient?
For COVID-related products or for other products?
For the next pandemic.
For the next pandemic. It'll be a new administration.
This is true.
I do think there'll be a lessons learned, and FDA has always had, really since 9/11, has had some form of a medical countermeasures emergency preparedness task force, but it has expanded exponentially, and I think there'll be a lessons learned, and they'll have people who, both from a science perspective, a policy perspective, and a legal perspective, will be ready to be deployed if something like this were to happen again.
One would hope. I hope so. I don't share your faith, but I do hope so.
Be optimistic, Vincent.
Yes. Denise, is there anything important that we should touch before we say goodbye? Did we forget anything? Is that a good overview of what goes on?
I think it's I think we covered most of the important issues. And I think that, it'll be interesting to see as things unfold, particularly on the vaccine side over the next few months. And I'm glad you all are talking about it every week.
Oh yeah. We talk about it all the time, and we're happy to have an expert. So we appreciate some clarity. All right. Denise Esposito, Covington and Burling. Great to have you. Thank you, Denise.
Thanks for having me, Vincent. I enjoyed talking to you.
Thank you very much.
Thank you.
This was really helpful.
Bye-bye.
Loved it.
Bye-bye.
Bye.
Kidoki.
Well, that was awesome.
That was very cool.
Fantastic. Yeah.
Brilliant, because the insights are great.
That's great.
And it's something that we just can't do at all.
Yeah.
We can do very little, actually.
But we do it very well.
I've talked to a lot of people at the FDA over the years. In fact, when she mentioned Janet Woodcock, I'm like, wow, Janet Woodcock has been there forever. She runs the place. But yeah,
sure, that's true.
But it's great to have that kind of an inside perspective and somebody who's actively involved.
Yeah.
Yeah, I feel a little better. I'm glad that if things aren't right, the lawyers are going to work on it, right? That's important. I'm glad to hear that.
My guess also, Vincent, if there's enough economic incentive to develop something, because there's this enormous market waiting for it, to get encouragement from the FDA is absolute gold, and you can convince investors and that sort of thing to buy into it.
My biggest concern, besides safety and rushing, right? I do not want political influence on the FDA. They need, as she said, they need to be able to evaluate the science. Well, look, the emergency use for HCQ got withdrawn. That's good.
Yeah.
So I didn't tear this thing up. It's still on my desk. It says that science has always been political. It's right here. It says so in science. So if FDA applies science to their decision making, then there's got to be politics.
Well, that
it's
statement.
important to remember.
There's going to be a rush for this vaccine. There will be a rush for
agencies. The head is often a political appointee, like at FDA. The director of the FDA is a political appointee.
Sure. So is NIH, right? Yeah.
But everybody who works there, all the people the rest of the way down the line,
they can slow it down.
they pretty much have made a career of this. And there are people like Denise who know what they're doing, who know how this stuff is supposed to be done. And some of the wingnut conspiracy theorists refer to this as the deep state. I prefer to think of it as the rule of law, which is a more accurate perception, because the people who have been doing this for a while understand what is legally supposed to happen at that agency, and they're going to do it right.
That's right.
One hopes so. Anyway, yeah, that's what it was illuminating. So thank you,
very, very
Denise. Couple of things before we do some letters. We have a postdoc available in the laboratory of Vinayaka Prasad, who is over at Albert Einstein College of Medicine, which is in the Bronx.
And that's a postdoc position. You're not actually giving away the postdoc.
That's correct.
Actually, he has two, one on HIV and one on SARS-CoV-2. And we talked about work from his lab on TWiV 567. So we will put a link to the job announcement here and check it out.
Cool.
And I was talking with someone. I guess all these interviews are going to be Zoom interviews, right?
They are.
Why not?
Wow,
Why not?
how different. How different.
My sister is on the job market right now, and
it's all Zoom.
Doing everything on Zoom.
I don't know.
Better have a good background. That's all I can say.
You could get someone to sit in. You could get Alan Dove to sit in for you and take your interview.
That's right.
And then you'd never get any job.
And we had
a number of listeners have sent us a link to rapidtests.org. Which is a cool site put up by Michael Minna and his collaborator, Larimore.
Cool.
With links to various things and graphs and information. It's very good, and you can subscribe, so they'll send you updates. And they even have a link to TWiV, his interview on TWiV.
Cool.
Very nice.
Okay, so that's cool. Rapidtests.org. Many people are sending it in.
He's got a great name, too, because his tests only take 1 minute.
Dixon de
That's an
Pommier.
Alan joke. That's an Alan joke. Yes, sorry.
We also have 2 new T-shirts. One suggested by Daniel Griffin. He said, we need a T-shirt. A TWiV T-shirts, masks are cool.
Right.
And so I asked Sharon Isern at Florida Gulf Coast University, who's a part-time artist. And she designed this one, which I really like.
It's very good.
Now, is this a Peter Max inspired design?
No, this is, remember the Bob Dylan poster that looks just like this? She said, I think, who was the person who did it? It was an artist, not Peter Max, though. But anyway, the original image was of Bob Dylan, and then she adapted it to this, which I think is nice because it's cool, right? The image is cool, and so are masks.
Yeah, and it'll get through to boomers, so that's good.
And then we have our listener, Eric Ebeling, who's a graphic designer artist. He sent us in last time his design for countering a miasma of anti-think. And I just said, why don't you use the real TWiV logo virus? And he did, and there it is. So they're both at cafepress.com/TWiV if you want to really geek out. Cool. It's very cool. I have to find out the name of the artist who did the Bob. Do you remember the Bob Dylan thing? Right,
I have seen it.
The
it's
iconic.
Not Peter Max. It's
here it is. It is. She told me. She said it's Milton Glazer's iconic Dylan poster.
Milton
Glazer. You want to see it? Come on, I'll show it to you. You might as well. And those of you,
I see it. I got
put
it
up. Put it up.
Here it is. Milton Glazer's iconic
there you go.
Dylan poster. And you can tell that's Bob Dylan, right?
You can.
She used my profile actually to make the thing, but you can't really tell it's me. It's just a generic blob. And anyway, they're both cool. cafepress.com/TWiV. It's not to make money for us. It is to show your love for countering a miasma of anti-think.
Vincent, may we all get masks with TWiV as the logo on the front, and then I want you to take a screenshot of all of us wearing it, because that would make a wonderful statement also.
You mean like this?
Yeah, I do mean like that.
Yeah, I'll send you all masks.
Send out all of us masks, and we'll line all up, and you can take a screenshot of the whole bunch of us.
As if Vincent didn't have enough to do. Vincent, mail us all stuff, right?
This will help.
Okay, that's no
many
problem.
ways.
But as I said, most of them have to do with opening schools, and we'll probably have an episode or two on that. But my feeling is, if you can't test, you shouldn't open. And it really depends on where you are, of course, if there are no cases. And that's what Tony Fauci said.
And I was very relieved to hear that my daughter's school is delaying their hybrid opening thing because they're clearly not ready for it yet.
Well, there are a couple of schools in Georgia that opened, and then they found positive students, and then they had to close again.
Yeah, it's going to happen. So, the first week you're going to have positives. Of course.
What they expect.
Well, the camp, they said you had to have a test in the previous 12 days.
The camp.
Anyway, here's one from Marissa. Dear Vincent and fellow TWiVers, I am not just a New York City public school teacher. I'm also just a scientist. Been a teacher in the New York City public school system for a little over 10 years. I came to teaching as a second career after having been a research scientist working as a molecular biologist and yeast geneticist, most recently on DNA dynamics at your institution. That would be Columbia.
Yes.
I did have a brief stint in a virus lab long ago, working on DNA packaging and T4 bacteriophage during the summer as an undergraduate student. That could actually date you. Right, Rich?
Could do.
I'm writing to you on behalf of New York City public school teachers, a group of teachers at my school, and she is at the Bronx High School of Science, is advocating for a safe reopening of our schools. My colleague and I have written a letter to Governor Cuomo imploring him to wait to bring students back into school buildings until it can be done safely. I think he just announced today that schools are opening.
Yes.
Incidentally, my colleague is also a research scientist turned teacher who worked at your institution in a virology lab, although we did not know each other at the time. She's now an avid listener of TWiV. A link to our letter is provided. I hope you can take the time to read it. I have personally read it, but the link is there and everyone can read it. I think the influence of TWiV will be blazingly obvious. We're in the process of distributing our letter to collect signatures in support of our cause before sending it to Governor Cuomo. We seek signatures of school faculty, staff, students, parents, alumni citywide, as well as any interested parties who have a stake in keeping the curve flattened in our area toward helping to ensure the health and safety of everyone. Given the lack of appropriate safety measures in place, we do not see how schools can resume in-person learning in New York City without triggering a second wave of infections. As an expert in the field of virology, and along with Michael Minute, an expounder of the urgent need for rapid, frequent testing, I hope you will sign our letter. We would appreciate anything you might be able to do. We are all in agreement that we must follow the best available science and do what is right in order to safely reopen schools. In the words of Daniel Griffin, give us our brakes and seatbelts. So we'll have a link to the letter. She also sent a little graphic, which has a code, a QR code where you put your QR reader on it. We'll bring you to the website as well. So we'll put that up and spread the word. All right. Thank you, Marissa.
And Marissa is Dr. Marissa Wagner in the biology department at Bronx Science.
Indeed. Dixon, can you take Mark's?
I would be happy to do so.
Because it has a haiku, I know you like to read the haiku.
I do. Thank you. Mark writes, Dear Team TWiV, here's another submission in your haiku arc. Fauci, the doctor, won't lie for the president. He deals with facts.
Really good one.
That's you should put that over the door.
What door?
Recently, quotes seem to have displaced chit-chat about weather. Here's another one from Yogi Berra. He foreshadowed this pandemic. It ain't over until it's over. Yeah, that's true. Well, in baseball, that's absolutely true. And weather, I'm not so sure. Weather, wear a mask, stay at home, wash your hands. And then there are two other things there that Tony Fauci would say. What are they?
Physical distancing.
Keep your distance out and try to stay outside as much as possible.
I do stay home for sure, although I'm not home today.
Yeah.
All right. Thank you, Mark.
Yeah.
Rich Condit, this next one's for you.
Anthony writes, mice do enjoy exercising in wheels. Oh, great. Exercise wheels are great for enrichment. The wheels will need to be swapped out and cleaned every so often. For what it's worth, Anthony, I just thought that was the best assay for whether or not a small animal is sick, whether they spend time in their wheel. Though, as you point out, you could photograph them and see how much they move around, Vincent.
That's a good idea. All right. Nice to know. Thank you. Alan Dove.
Sure. Tim writes, TWivers, it's been a very long time since Michael Mina's appearance. Yes, I mean that with respect to the message and call to action, low CT testing now. Since then, I've written my congresspeople, my daughter's college, my own CEO, and I reached out to E25 Bio with an offer to personally pay for $50,000 worth of assays to shop at cost on my own time to schools and elderly care facilities in my community.
Wow.
Nothing. Crickets. Clearly, I am not a person of influence, but more poignant is the lack of urgency and attention. I wonder if we might own up to the fact that virology is being used as a tool in the current environment to funnel money and support causes beyond public health. Clearly, those in executive positions in our own government are using virology to torture children at our border. If that sounds hyperbolic, it is not. It's happening now, using the pandemic as justification. At what point do we not start to consider that this crisis, this crisis not only has been unfortunate, but is actively being used to drive down civilization as we know it? I guess we will have to wait for the treasury bonds to go AA. Let's not stop there before anyone takes notice. In fact, the attack on the federal bond rating might be the ultimate goal here. Insert theory here. That's one I hadn't heard. There seems to be very little action to get virology, immunology, and science back into the net positive column as a force for good. No question here, just a comment. Virology is a tool. Okay. And P.S. My daughter just got her no school until 2021 if then letter this week. She started a job search only to find a U.S. Border Patrol position coming up in her search. Joel. Okay.
I don't know. Tim, some of the points you make are reasonable, but I think there's a lot of momentum behind the Mina approach. I'm seeing tons of letters and people are writing left and right, and I'm getting news reports, governors here and there.
It's been covered in major news outlets. I think it is getting attention, and I think and it's not, to be clear, Michael Mina is not the only person. I mean, he's not like some one guy who had this wacky idea. There are a number of people who are working in very close parallel, and some of them are working in relevant places like the FDA. And I think there's momentum building. So that this idea, he's been a major proponent of this and is certainly driving a lot of it. But don't pin it all on him.
And if people remember back to that episode, a lot of us were sort of surprised and blown away as he was talking about things. And, he was saying things that we hadn't thought about before, and he was making a convincing case. So realize that this isn't something that's necessarily obvious, and is taking convincing of people, just like it took convincing of us on that episode.
Yeah, there are the people who are proposing this idea. Michael Mina is doing the best job explaining it, and I think is really getting the word out there, and it is catching on, but it is not an obvious thing until you. It's one of those ideas where if he has 30 seconds to tell you about it, you'll think he's crazy, and if he has 10 minutes to tell you about it, you'll think he's brilliant. So it's the kind of thing where you actually have to wade through the introduction.
What's not clear to me is just how advanced the technology actually is. So there is this sort of platform for other pathogens, so it's a proven platform. And I'm sure that there are, it fits conceptually with what can be done with COVID. I'm sure there are prototypes, but I've talked to another number of individuals in industry and elsewhere about, and I've looked to see what's actually out there. And I don't think there's a whole storehouse or even a potential storehouse full of these things that fit all the criteria that are there waiting to go, but for the approval of FDA. I think my sense is that even technologically, we got a ways to go on this. In particular, making them as cheap as he says.
He implied he had some in his office that work.
Right. What I'm saying is that there may be prototypes out there.
Yeah.
Okay.
Yeah.
Yeah.
But does he have $50,000 worth to ship to this guy to take around to schools, and does he have another million of them to use for the school year? Probably not.
That's what he's trying to do.
That's what exactly. So that's the step we're at. So what Tim is writing about here, the idea of let's go do this now. Okay, great motivation, but not quite now. Maybe let's do the step of
today. The Times has an article about this just today for the first time. Okay, I would argue that TWiV played a big role in bringing this out because not only did the four of us sit and listen and go, "Oh my gosh," blown away, and people saw us in real time asking questions and getting it. And it's not just a op-ed that you put out there like he did. And it doesn't get much reaction because it's a bunch of words, right? And us going, holy crap, that's amazing. People that really resonated, and it's echoing. And I just think it's a big part of it. And, I'm sorry it didn't happen 3 months ago. I wish it had, but, he didn't push it, I guess.
Than never.
It's better late than never.
In anybody's estimation here, how close are we to actually achieving that?
I have no idea. It should be tomorrow.
Rich, you said you've surveyed the equipment and everything.
I've talked to some people in the industry who from their perspective, there are some kinks to be worked out of the technology. Getting absolutely the doesn't have to be absolutely the best, but the right antibodies on the strips and achieving what we're talking about, that it's not like it's right now ready to go. There's some development left to be done.
Well, I mean, that all can be done. That's not rocket science. And as you said a few weeks ago, Rich, man, we made an atomic bomb during a war.
The other issue is that when I talk to these individuals about $1 a strip, they say, that's pushing it. But on the other hand, as we've said before, there really should be between government support for this.
Between Gates and Bezos, they could pay for them all.
It ought to be free.
They have plenty of money. Come on. This is a war, folks. Alan, you probably want to go now, right?
No, actually, no tennis today because the tennis facility had no electricity, and there were thunderstorms rolling through, so they couldn't actually go outdoors.
Okay, very good.
Yes, I can actually stick around.
Brienne, you're next.
All right. Steve writes, hello, TWiV team. It is 23C with an 8.5 UV index in Carlsbad, California, like many days throughout the year. I just finished listening to TWiV 640, Test Often, Fast Turnaround with Michael Minna, and have been passing along the information to several friends and colleagues nonstop. The episode stimulated several ideas and a couple questions I wanted to ask the TWiV team. Actually, I was a bit surprised that there was initial skepticism on pooling, but I am glad the TWiV team understands its merits. We have been using pooling methods in high-throughput screening, HTS, for decades in preclinical drug discovery. It's a very effective strategy, and the deconvolution methods are well established. Pooling has been especially effective in conjunction with combinatorial chemistry methods that were developed back in the 90s.
Hey, Brienne.
Yes.
What does deconvolution mean?
I think it is used to look at your pool and figure out which one of those samples in the pool is positive.
I thought there might be some who might not know that.
That makes sense. I will say to Steve, I think that the deconvolution part of this was less where I was skeptical, and more of the dilution of a positive sample was where I was skeptical. That's where Michael Minna won me over on that one. Steve continues, I was so pleased to see that the FDA recently, July 18th, issued the first EUA for pooling up to 4 subjects. Do you think the spike protein density on the surface of SARS-CoV-2, i.e. the number of spike surface proteins per virion, might be related to the virus's infectivity and virulence? How about the speed of immune response and sustenance? I appreciate these probably fall into what Fauci and TWiV 641 characterized as data-free zone questions, but perhaps the TWiV team can speculate. Along those lines, I'm wondering if RNA repeat coding for spike have been evolving that may result in increased spike expression, perhaps overlooked in basic PCR given the number of copy runs issued. I am wondering if it is possible to quantify spike density through the analysis of data across PCR cycles within a typical diagnostic run, i.e. without full strain sequencing. I would expect more spike-rich SARS-CoV-2 may improve test sensitivity and perhaps enhance the kinetics, i.e. speed to result. This may help change community mindset and facilitate approval against FDA pushback.
Hold, stop before you go on. There's too much stuff here.
Yeah.
I don't know why PCR would have anything to do with spike density.
It sounds to me like he's thinking that there might be gene duplications in the genome.
I've never seen
that doesn't happen.
No,
that doesn't happen in an RNA virus.
Right. So the confusion here is the spike protein density on the surface of the capsid would not be controlled by the number of copies of the spike gene present, and the virus doesn't have enough room in its genome to duplicate genes. I mean, we see gene duplication events in the human genome because we got lots of space. But the PCR is looking only at the genome of the virus, and that's not the way you see it.
And pox viruses, right? Genomic accordions.
But we did do a paper some time ago that was looking at the effects of this, what is it, D614G mutation, or the other way around, a mutation in the spike protein that had the effect of forging a stronger association between the two subcomponents of the spike, so that the density of actual active spike protein on the surface of the virions was higher in one virus than another. And they showed that in that virus, it was more efficient at infecting cells. Okay, so a higher genuine spike protein density on the surface in those experiments is correlated with, in culture, a better ability to infect cells. Whether there's no evidence that correlates with any kind of virulence or transmission or anything else. That's just a laboratory experiment.
Yep.
So here's what I'm thinking. So different viruses have different spike densities on their surfaces. And in particular, coming to mind, the HIV spike density is very low, which makes it hard for neutralizing antibodies to bridge them and neutralize. But, influenza has tons of spike density, and, it's neutralized well in some cases. So that may be related to that, but I don't know that we know much about it here. It's probably here. I think it's from what I've seen of the reconstructions, but I don't know how, if you look at the EMs, it seems to be intermediate between HIV and influenza in terms of spike density.
Yeah, so I think that the stuff he says at the beginning of that paragraph about spike protein density having a role in biology might make sense, but I don't think that's something that needs to be taken into account in the diagnostic test, especially when we're using tests for genome.
And I think spike density can be a little difficult to determine accurately because it's somewhat fragile. So if you want to purify a virus or do anything to it to measure the spike density, you're not necessarily seeing what actually originally came out of the cell.
Right. So, we have to get Jason McClellan on because he's into this sort of thing.
Yes, absolutely.
He, for example, makes pseudotyped VSV with different spikes, and, they assume the geometry of the original virus on the pseudotype. The way the spikes are arranged and so forth. So apparently it's built into the spike protein sequence. And even if you put it on VSV, it still looks like the original virus. So I bet he would be into this.
Mm-hmm.
Okay. Go ahead, Brian. Sorry.
All right. Many years ago, I had the privilege to work in an exciting company in Silicon Valley, Affymax. Affymax had many successes, including developing the gene chip technology, now the famous Affymetrix. Alexandro Zaffaroni, Alex, was the founder of these and several other impactful companies. Alex was legendary for orthogonal thinking. He constantly sought to join many disciplines to solve complex problems. Alex understood that many drugs came from natural product screening, but one of the greatest challenges was sample collection. Prior to his passing, Alex once told us years prior he approached the then CEO of Coca-Cola, knowing that Coke had one of the largest worldwide distribution networks. Alex pleaded with the CEO of Coke to collaborate. If Mr. Coke would be open to instruct all its drivers to scrape the bottom of their shoes into sample envelopes upon return to their hubs and sent to one of Alex's companies to screen, Coke would in turn get a percentage of every drug discovered developed. Unfortunately, Coke passed on this multi-trillion-dollar opportunity. Now you should talk to people like the Tiny Earth Initiative who are doing that with undergraduate students, collecting samples around their community.
Well, there's a suggestion in the next paragraph.
When when we used to go, my wife was at Merck. We used to go on vacation. She would collect dirt from wherever we went, put it in bags, geotag it.
Mm-hmm.
You know, all the people at Merck when they went away, they used to do that. So
just ask Bill Campbell about that.
This reminds me of this is on a much smaller scale, but the original luciferase assays.
Yeah.
You got to be really old for this, right?
It's true.
Sigma would sell you firefly tails in a bottle.
Really?
Okay,
That's right.
Because I bought them, and you grind them up and get luciferase out of them.
That's
and the
right,
way
that's
they
right.
had the way they got the firefly tails was they paid kids to go out and trap fireflies.
Yes.
Sure.
And they would give the kids a certain amount of money for fireflies, and you get a cap that said that you were part of the Sigma Firefly something club. Or something.
Nice.
I had caps.
Illumination program.
I bought them for everybody in my lab.
Firefly extinction program.
Nice.
That is, as both a kid who like to catch fireflies and someone who uses luciferase often, I love that. I want one of those caps.
That's true.
They might still sell them.
eBay.
I will be looking into that. So Steve continues. TWiV seems to have a very broad reach. Maybe there is a listener out there with a relationship to Jeff Bezos and might pass along a similar thought. Perhaps Jeff is listening himself, or should. Amazon delivers to and picks up from a huge number of households. Amazon is well poised and could readily deliver a sample saliva vial to each, especially prime customer household, and collect back the pooled household saliva sample. Many of us receive deliveries multiple times a week, especially now, so a short time series is also possible. Coupled an additional pooling strategy across households and communities with inexpensive paper tests at a few central testing locations, we could rapidly identify burgeoning COVID hotspots and households in a matter of days. The federal government fumbled the coordinated testing ball, but a couple companies and the richest person on the planet with an impressive infrastructure at his disposal can help recover the ball to help us out of this mess. Steve.
It's true. Companies like them and FedEx, and they can do this if they want to.
They can.
I found the hat.
The tests become available.
I found the hat, Brianne.
Oh, excellent.
$17.99.
Where is it, on eBay or somewhere else?
What's it called? I don't know.
What does it say? What does this hat say?
I'll look for it.
Sigma Firefly Scientist Club. It's got a little badge on it.
Cool. All right. Well, Steve is the CEO of Idogen Certainty, Inc., which Kathy has written. It's a drug discovery software company.
Cool.
And I know it's Kathy, even though she's not here, because it's written in purple.
It's written in purple.
All right. Mike writes, rapid testing gaining attention. Governor DeWine in Idaho just referred in his press conference to the need for rapid point-of-care COVID tests. Additionally, he announced that Ohio was entering into an agreement with several other states, Maryland, Louisiana, others, to secure these tests, to create demand for these type of tests, and he provides a link to that story. Mike Toledo, great. Excellent. Yep. Dixon.
Rebecca writes, hello from Toledo, Ohio, where my second son was born, by the way, where it is 74 degrees F and 23 degrees C. I was just listening to today's press conference given by Governor Mike DeWine, and I must admit that I geeked out when I heard him announce the following, and that was his announcement about the rapid testing. He quotes, today I'm announcing that we are entering into a multi-state purchasing agreement with Maryland, Louisiana, Massachusetts, Michigan, and Virginia to expand the use of rapid point-of-care tests. This will help us detect outbreaks sooner with faster turnaround time, expand testing in congregate congregate settings and make testing more accessible for the most high-risk and hard-hit communities. Scaling up the use of these rapid point-of-care tests will serve as an important screening tool and a critical addition to our plan to limit the spread of COVID-19. It certainly appears to me that the effort on the part of TWiV listeners to get the word out is having an effect. Officials are listening and taking action. Sometimes it feels that everything I am able to do is too small and insignificant to make a difference, but knowing that I, along with so many others, played a part in raising this topic gave me a much-needed glimmer of hope. I am just an archivist working on my master's in library and information science, but the letters you've read from other readers underline just how much the population needs a stronger understanding of science in general and virology in particular. Thank you for giving your listeners such wonderful ammunition for striking back against the miasma of anti-think. Rebecca.
Yeah, Rebecca, you can wear that shirt.
Yep.
There you go.
White or gray. So it's available in white or gray.
By the way, in case you're wondering, Toledo is the 9th largest port city in the United States.
Oh.
And they're the glass capital of the United States. They make plate glass like crazy.
They should not throw stones.
They should not.
Rich Condit.
Chris writes, my name is Chris. I'm a UCSD alumnus, drug discovery manager for a CRO and founder of a startup here in San Diego. I've been reverting to your podcast as my main source of BS-free COVID information. I know that we're entirely BS free, but it's high quality BS, and have turned all of my friends to your channel as I have heard one too many false facts spreading through our friend group. I have a slightly peculiar question. If you spray down a surface that's been inoculated with virus after this is good with the virus after. I'm sorry. If you spray down a surface that's been inoculated with the virus with a saline solution or ocean water, would it damage, kill, or diminish the viability of the virus on that surface? Why I'm asking? My startup, Surf Up, is an automated surfboard rental station. We'd like to ask customers to wash down the board in the ocean before bringing it back to the station, but I would like to know if that is a viable solution. I've been searching for a research article, but haven't found any studies looking at the viability of the virus in a saline solution. Do you know if any such studies have been conducted? Thank you for your prompt attention to this.
What a great idea. You know, here in New York, we have bicycles in racks you could rent.
That's awesome.
And there in California, you get surfboards.
Rentals at a surfboard, yeah.
It's great.
So
out of a vending machine.
My first reaction to this is that we always stored our viruses in a saline solution.
Yeah, yes. Regular
Right?
I thought you were going to say something about where you stored your surfboards.
And I would think that generally, in terms of survival, viruses might actually be better off in saline solutions than otherwise, though I don't know specifically about ocean water, but I believe the salinity of ocean water is not far from PBS.
Well, there you go. Kathy put it there for us, 3.5%. PBS is 0.9%. I don't know if that would inactivate virus.
So,
no one
yeah,
really.
so ocean water is much more saline than PBS, but I have not heard of salt being used as a
I think it won't work because there are many in 1 milliliter of ocean water. There are several million virus particles of all sorts.
Correct.
So I don't think your ocean water will work.
Well, but
it's working very well.
Right. What I would say is if you're renting surfboards out of them, bear in mind, this is a respiratory virus. And so a lot of the stuff you see going on with we're going to close the school on Wednesday to disinfect surfaces and we're going to wipe everything down. And after you've rented this, you wipe it down with the disinfectant. It's hygiene theater. So it's like the security theater we got after 9/11, a lot of which doesn't really accomplish anything. So I think in the case of a surfboard, I mean, what are you envisioning as the mechanism for that being the fomite for getting SARS-CoV-2 into somebody's nose? Because if you're inserting a surfboard in your nose, you probably got other medical problems that are gonna trump
I think
this.
you're doing it wrong.
I think you're doing it wong.
Yeah, so the washing of the water is probably
when
taking it off.
the water washing over it, and it's a smooth surface. It's I really don't picture the droplets, any kind of a moisture that this virus is going to need to survive being on that surface out there in the California sun for more than a few milliseconds.
Besides, you got to look at.
If it'll make your renters feel better, you could ask them to rinse off the board in the salt water before returning the board. I don't think it's going to make a difference because I don't think there's going to be a virus on there anyway.
I think in this particular case, the benefit-risk ratio in favor of surfing is really high.
Yes.
Okay. Nice question, though. Love it.
Yes.
So speaking of the BS part, Rich, you asked a question about dendritic cells on the last episode. And one of my former students sent me a text message about my answer. And it was a text message, but I can sort of imagine the tone as being Langerhans cells are macrophages, not dendritic cells. So just so you know,
okay.
I was corrected by text yesterday.
Alan Dove, you're next.
Sure. Tom writes, Dear TWiV, in a recent episode, you briefly mentioned that Maine is doing well. Living here and now watching license plates from places like Florida, Tennessee, Georgia, and even Texas appear with increasing regularity, I've been amazed at the low numbers we have here in Washington County, Maine, only 9 so far. Is this true? I watched the unmasked visitors pack restaurants and grocery stores as if we were living back in 2019, and am amazed there's been no increase in numbers. Today, I saw an article from the Bar Harbor area that mentioned
there you go.
The hospital there is seeing cases, but the numbers that get reported are correlated with the state of residence and not the state of testing. I could not believe it and went to the Maine CDC site. It is true. Quoting from the site, I think, who is in the data? Data are about individuals who claim residency in Maine, regardless of what state they were tested in or where they were currently living. For example, an individual who claims residency in Maine but lives in Florida will appear in these data even if they were living in Florida at the time of illness. County listings are by residence of patient, not location of the hospital or testing location. I then looked at the cumulative confirmed cases statewide and found the number 3,548. However, the same site states that there were a total of 5,385 positive tests.
Duh.
Based on this information, we could have 100 positive testing tourists actually in Maine who would not be recorded as positive tests in Maine.
All right.
How do we or those doing infection control determine if our community includes a rise in positive tests with this type of reporting? Thoughts, concerns? So I certainly have concerns. The first thing I would ask is, what is your state's interstate quarantine policy? Because Massachusetts right now has billboards flashing on the highways and they're stopping people at the airport and train stations and bus stations and making them fill out forms that if you have arrived from out of state, you have to present a negative SARS-CoV-2 test from within the past 7 days, I think it is, or you have to quarantine for 14 days after arrival in Massachusetts. So I imagine this is effectively shutting down the tourist industry in places like Cape Cod and Martha's Vineyard, and that's not so great because that's kind of how they make their livelihood in those places. But if Maine is not doing something similar, so Massachusetts has exceptions for states that have done well in managing their outbreak. Maine is currently on that list of exceptions. But if Maine is not quarantining people who come in, I think you may find yourselves off that list of exceptions sometime soon because there's some stringent requirements for the Massachusetts quarantine. And this thing with counting people based on the state they say they live in, that's concerning.
Yeah, for sure.
Yeah, it is.
Brianne.
Owen writes, Hi, TWiV team. I am soon to be a PhD student at University of Wisconsin-Madison with a master's in virology. I'm writing today to comment on something that hasn't been brought up in regards to rapid testing. Like all of you, I think that rapid testing is the answer to helping us safely weather the pandemic while waiting for a vaccine. It's been brought up that this test would be used in homes to test children each day before they go to school. If they're positive, they stay home. However, I don't think that this would always be the case. I don't have kids myself, but I can give an example in the food industry. I've known lots of wait staff who have been diagnosed with flu or other illnesses, etc., but still went into work because they'd be fired if they didn't, or because they couldn't pay the rent if they didn't go in. I think it's very likely every one of us has been served by a sick person at a restaurant. One of the reasons I think it's a bad idea to go to restaurants during this time. I think that some parents may be in this boat, and even if they aren't sick themselves, they can't stay home with their positive child because they may lose their jobs or may need the hours simply to pay the bills. Considering those in lower socioeconomic standing have already been hit harder by this pandemic, this could add an even greater strain. I was wondering if you all could comment on what you think could be done to mitigate this, if anything. Thank you so much for your wonderful podcast. You guys make it so much easier to stay on top of the deluge of information. Your perseverance in educating the masses has helped me keep my cool many times while being mansplained by conspiracy theorists how this pandemic is a hoax. Thank you for all that you do, Owen. Owen, I agree that being mansplained is really annoying. And I hope that you look out for.
Well, actually, Brianne,
we have
and I hope you look out for one of your classmates, actually, who is from my lab. So be friends with Tom. And I think you make some really great points about the additional challenges in having kids stay home from school or having people who test positive themselves stay home from work. And those things should be addressed.
I think there's got to be, if you're going to have some sort of at-home testing thing, there's got to be some way to verify the test result. Okay. As an app, there's got to be an app for that, right? To verify a test result and to clear you to attend school. Now, obviously, any of these things can be scammed, but something like that, at least to try and minimize.
Yeah.
I think the problem is obvious. Okay. To try and minimize that problem, there's got to be some sort of procedure.
I think there are actually two problems here. One is the potential to scam the test. And I think the way around that is not to do it at home, but to do it at school.
Yeah.
So you come into school in the morning, you spit in a cup, and by the end of first period, if you're positive, the nurse calls you out of class and you got to go home.
Yep.
So that takes that out of the equation. The deeper problem, which I have zero confidence that our nation will fix in the current environment, is this situation where people are being told to send their kid to school, and that puts them in an economic jam. And it really highlights, I think this letter really highlights the kinds of the regressiveness of a school reopening policy that doesn't take into account this issue. I mean, people who have the privilege of being able to school their kids at home can do so and avoid all of these risks. And maybe they've got a stay-home spouse or a work-from-home spouse, who can take care of that duty, and it's not a problem. Whereas the poor folks who have to send their kids to school because that's the only daycare they have are then placed in an impossible situation. Do you want to risk the lives of your family members by sending your child into an environment where they have inadequate infection control? Or do you want to risk your economic livelihood and therefore long-term health of your family by keeping your kid home and trying to homeschool them, which gets you out of a job?
And that also, ties into what happens if your school goes online. Can you stay home with your kid? I think that people should acknowledge the economic issues with that as well.
Yeah.
This is in the area of non-virology.
Yes, it is very much in the area of non-virology, but it is being driven by a virus. So
I understand. I don't know the solution. And people write and say you should just talk about what you know about. So I don't know anything about
the solution is that you continue paying unemployment benefits.
Well, yes,
to everybody
but
who can't hold down a job because their kid can't go to school.
I agree with that, but I don't have anything to do with that, obviously. But I agree.
I know, and that's why given the current environment, I have little confidence that we're going to work that out in this country, even though every other industrialized country has done so.
I agree with you. I do agree. Let's do one more round. Musashi writes, "Hi, I'm Musashi. Just want to ask how much testing per capita is considered to be needed to say that the number of COVID cases and the number of deaths is somewhat close to the real number, and how likely is that a country with very low testing is underreporting? For example, the USA has 1.35 more times deaths per capita than Mexico, but USA performs 23 times more tests per capita than Mexico. The difference is giant and opens the possibility that Mexico's actual per capita numbers may even be worse than the US. But can very low testing cover up the data? Do you think states within the US may actually decrease testing as a political tool to lower their actual per capita numbers?" No, that would never happen.
Do that. How could you think such a thing?
Of course. Finally, I just want to tell you that I really like you a lot and I appreciate you, and you deserve more views because your podcast is so perfect, and I hope you're having a great day. That is just great. They like us.
That should be our last letter.
Mushashi.
Although I'm going to read the next one.
Is so perfect. Alan, you say it should be.
Yeah. So a good indicator
5%?
of this is positive test rate.
Yeah.
Now it's still possible to scam this, but it becomes a lot harder. And in fact, one of the things, again, bragging about my state, that Massachusetts is doing to determine if a state is on the okay list for travel, is they have to have a positive test rate below 5%. So you can't just under-test your way out of the pandemic, because if you're only testing the people who show up in the hospital coughing their lungs out, you're going to get a positive test rate that's much higher than 5%.
Right.
Now, you could get around that by running out and finding a bunch of healthy people to test who you're pretty sure are negative, and then jacking up your denominator that way. But it would require a really deliberate effort to do that. In most cases, what's going to happen is if the state is just saying, we don't want to test so much because we get too many cases, what's going to happen is that the sickest people are going to get tested, and your positive test rate is going to show that. And in fact, that's what you see internationally in countries that are dealing with immense death rates from this, digging mass graves and that kind of thing, because they're not testing and not reporting a lot of cases. You see that the positive test rate is fairly high.
Dixon.
Yes, sir. Anne writes, "Dear TWiV, I am a 77-year-old retired carpenter living in Amherst, Massachusetts, where the temperature has been too damn hot all summer. As a non-scientist, I have really learned a lot from TWiV. I'm determined to learn about COVID directly from scientists and not from journalists or social media. Thus, my appreciation for your podcast. It is my understanding that it is non-symptomatic and pre-symptomatic COVID carriers who are shedding the most virus and therefore are the primary infectors of others." You want to stop here and address that, Miss?
Well, they're all shedding virus. Pre and symptomatic are all shedding, but the problem with these two is that they're silent, so they walk around transmitting. Yeah.
Bingo. I assume the non-symptomatic people who never develop COVID are unaware that they're shedding the virus, unless, of course, they are tested. Do you have any idea of the number of those people who never know they were shedding the virus? Another point.
About 30%, I think. That's
Thank
what
you,
comes
Rich.
to mind.
Yeah, something in that area.
That's current estimate, yeah.
Yep. Is the 2-week quarantine irrelevant for such carrier shedders?
No.
I.e., if with no further explanation, i.e., if they quarantine for 2 weeks, will they be past their shedding period?
Yeah, 95% confidence intervals. Yeah.
Okay. I'd also appreciate it if you would talk a little more about masks. It seems to me most people think they have a mask on. They don't need to maintain a 6-foot distance from each other. But since most of the masks don't seal around our mouths and nose, it seems to me we need to maintain 6 feet with the mask on. And if we want to take our masks off, we need to get at least 8 or 10 feet away. Is that correct?
Yep.
Yeah,
Yes.
you got it.
And I brought this up as early in the mask discussion. I want to point this out.
Yeah.
Listen, we should do all our letters like this. Also,
The risk compensation problem. Yes, don't do that.
okay, fine. Also, I have been keeping a mask in my back pocket and pulling it out on whenever I need it. If a delivery person comes or if I get close to some other person when I walk. Then back to the pocket it goes, along with whatever virus it might have picked up. The next time I'm in the house, I usually wash my hands. But is this bad mask practice? Or if I wash the mask once a day, is that okay? Answer, group. Greek chorus, please.
I think you're fine.
Yeah, I think
that
you're fine.
sounds similar to what I do.
Yeah.
Matter of fact, that's what I do, and I do. There's one thing I do that I don't know how important or effective it is that when I fold the mask to put it in my pocket, I fold it so that the outside thing is on the outside. Okay. And I make sure I wear it. I make sure I wear it in the same orientation every time.
This is true confessions.
The ideal situation would be if everyone had a little case with a UV light in it. You put the mask in it.
Perfect.
But we don't.
No, we don't.
I do the same folding thing, and I've been thinking of it as a superstition. Like, okay, I know this doesn't make it, but I'm doing it. I'm
so this becomes true confessions here.
That's right. Absolutely.
Of the TWiV team. And I should also point out,
we believe in science, but we don't only follow science.
That one of the most important things about the mask is keeping from spreading it to somebody else.
Yes.
And so from that point of view, it doesn't matter what orientation you're wearing it in or how dirty it is.
It's mainly to protect other people.
Yeah.
Correct.
And
lastly,
and it doesn't have so much of an effect that you can then get close to them. Yeah.
True. Lastly, I'll just comment that the University of Massachusetts is in Amherst, and we are all about to be inundated with thousands of students living both on and off campus. I am COVID scared. I've been diligently trying to get the powers that be to talk with Dr. Michael Mina. Thank you so much for your podcast, Anne. Well,
you learned your stuff. Hammering. You learned your stuff, Anne. Good job.
Yep.
Mm-hmm. She hammered her points home.
Yeah, you should stay home. Okay, don't mingle with the students. I guess.
Yeah. So UMass, I just went to their site to confirm this news. They have revised their reopening plan.
Right.
And
they're all
in looking at the national situation, they will make a major reduction in the size of the campus population. And I don't have all the details, but they're moving a lot of stuff online is the upshot of this. And I don't know if that's going to be everybody, but hopefully, hopefully that's what they'll do.
Right.
Rich, you're next.
Howard writes, Dear TWiV Mavens, I thought you might find the following emails about Mina's contagion testing proposal worthwhile. Calfonso Isemunik and K. Grant are all reporters for the Globe and Mail, education, science, and special assignments, respectively. Sam Hammond, Leslie Wolf, and John Weatherup are the most visible leaders of education workers unions in Ontario, elementary, secondary, and support workers, respectively. I hope that I didn't too badly distort Mina's proposal for your position. It would be good if you could put it up on your website. Put up on your website 2 standard emails describing, promoting Mina's proposal that could be sent by TWiV stalwarts. One to the reporters, the other to education union leaders. More than anyone else, these guys are likely to treat the proposal seriously and be able to motivate political leaders to act. Other standard emails to school board leaders, local public health officials, and relevant politicians would also be helpful in spreading the word. Most people find writing an email intimidating. Having a standard email as a place to start would increase the likelihood that the idea gets proper attention. Finally, there has been some coverage about the Canadian government's dismantling a year ago, for strictly bureaucratic reasons, the Global Public Health Intelligence Network, much like the U.S. government's dismantling of its pandemic preparedness group. When you decide to take a rest from the reading from reading the latest preprint research, I'd really like to hear an episode on the value of these two groups and those in other countries and NGOs like the WHO in alerting the virology community about the signs of potential new outbreaks. What sort of early warning systems should be put in place now? Thanks again for your work, Howard.
Well,
And
I put Howard's letter on the page, microbe.tv/TWiV/testing-letters. And if someone wants to get a crack at making two letters, they're welcome to. I don't really have the bandwidth to do it.
maybe whoever is doing Michael Minna's page can get on that.
Okay. Alan Dove, you're next.
Okay, Patty writes, Dear TWiV Troop, I'm new to the world of podcasts, but am now almost addicted. As a letter carrier for the United States Postal Service, alone delivering mail in a truck all day, I find it helps to pass the day. I love our audience. This is great.
This is great. So, and a postal delivery person can handle 2.5 hours of TWiV.
Yes, exactly. I tripped over the sound of Vincent's
voice
in mid-June 2020. I tripped over the sound of Vincent's in something voice in mid-2020. Have been an avid listener ever since. The first time I heard Vincent in June 2020 was on a podcast that originally was aired on May 15th, 2017, called Every Little Thing. The episode was titled The Quest to Wipe Out a Virus. Wow. Vincent was obviously an expert speaker on the episode. I was so intrigued, I Googled Vincent's name, and here we are. I listened to the June 14th TWiV episode called Fancy Ferrets, and now I cannot get enough. Even went back to the January 12th and 26th episodes to hear some of the first conversations about SARS-CoV-2. I have no school-age children in my world at the moment, but I'm still concerned about the reopening of schools across the country. My question concerns face coverings. When face coverings are being discussed, we're often talking about face masks. What about clear plastic face shields? Can they give a teacher the same or even better protection than a cloth, mouth, and nose covering? Seems to me the younger students especially, it's vital for them to see the teacher's face in its entirety during instructions. As I understand it, vital pathways are still developing in a human's brain in the early school years. I just do not hear much discussion about the use of face shields, and was curious if this is an option. Thankfully, virtual learning is an option. At least it is something. I'm not as worried about as others are when it comes to children wearing face masks. Children spend a great deal of their day following instructions, and I think will be more compliant than the adults. In fact, I bet the children would take pleasure in correcting an adult about the proper usage of a mask. I think face shields probably ought to get a lot more discussion.
I think there's been some data that has come out that showed that they were not as effective as masks. Okay. If they are sealed, then they fog.
Right.
And they are effective. If they're not sealed so that you don't get the fogging, then you also are getting a fair amount of
and by effective in this context, it's important that we point out we're talking about laboratory analyses where they do these laser illuminated spray type of things, not actual public exposure things, because we just don't have those data on the cloth masks. Okay. It's obvious that an inexpensive rapid at-home test would be the best course of action as described by Michael Minna. I've written to my Florida state representatives about the rapid test, and the only response I received was auto-generated. I even wrote to Dr. Sanjay Gupta and Chris Cuomo of CNN with hopes that someone would read it. Why isn't this being explored more? I wait with bated breath each day to hear about it outside of TWiV. I had no idea this science would ever interest me. When I hear things on the news regarding the current pandemic that concerns me, I'm anxious to hear what the TWiV Troop has to say.
Cool.
Yes, thank you for countering the miasma of anti-think. Brilliant. Although some of what is discussed on TWiV is beyond my abilities to understand, I do learn more each time I listen, and now also watch on YouTube. Keep up the one. Just don't watch on YouTube while you're driving the postal truck.
Yeah,
keep up the wonderful banter. I appreciate you having all in my ear each week. Also, I almost forgot, it's partly cloudy here in Port St. Lucie, Florida. It is a typical 87 degrees Fahrenheit with a heat index of 99. There's a 20% chance of precipitation. We just said goodbye to Tropical Storm Isaias, which is now hitting the upper East Coast as a hurricane. Patty is in Florida.
I love this. I had no idea the science would interest me. And that's why I tell people, just try it. You just don't know until you try it. Don't assume just because it's science that you're not going to like it.
Yeah.
Actually, that brings me back to a discussion we had a couple of episodes ago about somebody, calling us, I don't know, experts or distinguished or something weird like that. Okay. Well, the point I was trying to make is that you can do this. Okay. It's mostly just learning how to critically analyze stuff. Okay.
It's true.
Yeah. We know a few facts that we've accumulated over time, but you can do the same thinking. And you can do the same thinking we do.
It's
There's
just
a lot
about
of
asking
people.
questions.
A lot of people out there that love to solve puzzles.
Yeah.
Science is solving puzzles.
Absolutely.
And by the way, Patty, some of it's above my head too.
Yes, you hear us getting in experts and asking questions and saying, I don't know, and please. And I'm constantly defaulting to Brianne, like, all the immunology stuff. Give me a break.
And the other day, Brianne said, I don't know everything.
No, not at all. There are many, many, many cases where I am a little worried sometimes when we talk about an immunology paper, and I'm like, boy, I hope they don't ask about this part.
Ooh,
And yet they do.
Sometimes,
there
yeah.
was a while back. There was an XKCD cartoon that had a graph of things I'm good at and the universe of things that I'm not good at.
Mhm.
And it pointed out that to a first approximation, everybody sucks at everything.
That's great.
Average.
That's great.
I mean, yeah, we know a lot about a little slice of the universe, and, and yeah, join us.
When I first moved into where I am now, our neighbors came to visit us. I said, "What do you?" They were both lawyers. And the guy said, "What do you do?" And we both said, and he said to us, "You guys know an awful lot about nothing." And then I told my wife, I don't want to talk to that guy anymore. I never saw him again. All right, one more email.
But suddenly the things that we know about are not nothing anymore.
Yes.
Righto. Brianne, take that last one.
All right. Peter writes, "Hi, TWiV team. Greetings from Antalya, Turkey, where current temperatures are in the mid-30s Celsius in the day and mid-20s Celsius at night, with humidity ranging from 33% to 66%. The COVID statistics in Turkey are not looking too bad at the start of August, though we are wondering what the effect of opening hotels to overseas tourists will have. Some personal concern there, as my stepson has just started to work at a hotel." And he gives a link from Worldometer with Turkey's statistics. "The use of 80% alcohol lemon-scented cologne hand sanitizer has been common during practice for over 100 years in Turkey. It is an important tradition to offer cologne during guest visits, on bus trips, and in restaurants. Its offering during holiday family gatherings has also become somewhat of a ritual. If you should visit a Turkish house, the first thing you will be offered is cologne to freshen your hands." And he gives another link about that. If you are, again, taking listeners' picks, I have a link to a topical Second World War British film about how coughs and sneezes spread diseases. That rhymes. It includes interesting suggestions for various homemade masks. This was from the Ministry of Information and produced by Verity Films. And he gives a link. Regards, cool. Peter.
Sure, we'll take a pic.
Yeah, and I think that this practice of frequently using hand sanitizer is a pretty good one.
Well, yeah, if you sent it with lemon, that's great.
That's a great idea.
I like that. Some of the other scents are not so good. There was when my kids were young, they used to watch Thomas the Tank Engine. And I remember there's one part where one of the engines said to the other, "Coughs and sneezels spread diseasels." They were diesel engines, right?
Yes.
Yeah.
Right.
I'll never forget it. All righty. TWiV 651, show notes, microbe.tv/TWiV. Questions and comments, twiv@microbe.tv. And if you'd like to support us, microbe.tv/contribute. Brianne Barker is at Drew University and on Twitter, BioProf Barker. Thanks, Brianne.
Thanks. It was great to be here.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
Dixon Despommier, trichinella.org, livingriver.org. Thanks, Dixon.
Always a good time.
Alan Dove is at turbidplaque.com. Alan Dove on Twitter. Thanks, Alan.
Thank you. It's always a pleasure. Good to be here to the end, too.
Thanks for staying.
I don't know. It doesn't sound right. Good to be here to the end.
Good to be here to the end.
It's like a sinking ship.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.